Hi everyone, good evening, good afternoon guys. How are you all? So guys, welcome back. This is Tina, your biology mentor, and this is Re-NEET Bounce Back series. And before we proceed guys, please remember that days are like the countdown has really begun, and now you really need to practice things on daily basis. You have to practice questions. So you have to take the test so that you set your uh, set your mind in that zone. So yes guys, so for that we have Unacademy All India NEET test series, right, with complete syllabus coverage, detailed result analysis. You have to take that just for rupee 1, but it's not about money, it's about your practice guys. You have to practice these questions day and night. Now take these sets. Good morning. Okay, let it be morning. Last 4 days, so the wait is almost over. I know it's now adrenaline rush, but honestly, we were really waiting for this day, weren't we? How many of you are actually waiting for this day now? Let it be over. We are well prepared. Let this be over. How many of you are ready for this and waiting for it? Yes, let's start, guys. We will start. How many of you are waiting for it? Let me know. Me, Haripriya is waiting. Namrata is waiting. Wow. All right, guys. Let the wait be over. Very soon it will be. But now we have to be double prepared. We are double prepared, and all the last moment revisions are going to take place now. So make sure that you stay tuned to this channel for all the notifications as well as for the last minute revision, right? So let's revise the questions. Yes. Quickly like, subscribe, share the session, guys. And we are going to start with questions. All right. So starting with chapter number 1 in living world, guys, we really don't have any table here except, sorry, we don't have any diagrams in here except for this one table which shows us the ascending order of taxonomic categories. It starts from species and goes up to the kingdom level, right? Then this table you should be done with that. What are the biological names, the genus, the species to which it belongs, right? Wait, I move a little bit on this side. Yes. So they've repeatedly asked the question on housefly, Musca domestica, family is Muscidae, order Diptera, insecta arthropoda, man is Homo sapiens, Homo is a genus, hominid, D is the family, order Primata, class Mammalia and Chordata, mango, Mangifera indica, Mangifera anacardiaceae, sapindales, dicotyledony, and then wheat is Triticum aestivum, family is Poaceae, also known as Gramineae, then poales, monocotyledony, and both mango and wheat are angiospermae. Are we clear? Are we clear? All right. So with this, the first chapter is done. Now moving on to the chapter number 2. All right. So chapter number 2 is, guys, biological classification. Let's see here. The first table here is characteristics of 5 kingdoms. You should be very thorough with this table because the questions are direct. Cell type. Out of cell type, only Monera is prokaryotic, while rest of them, they are all eukaryotic, right? And what is the cell wall of prokaryotes or Monera made up of? It is non-cellulosic, that is made up of polysaccharide plus amino acid. What is the name given to this? Peptidoglycan. Made up of polysaccharide plus amino. Audio, video, absolutely normal. Yes, guys. So talking about protista, present in some. So we know that protista is a connecting link between all the other kingdoms. So in some protists there is cell wall while others don't have. Fungi will have cell wall made up of chitin, plantae will have cellulosic cell wall, and animals don't have cell wall. Nuclear membrane is absent only in Monera, that's what makes it prokaryotic, and rest of all will have nuclear membrane. Then talking about the body organization, see, Monera and Protista have cellular level, and then fungi, multicellular, loose tissue, Plantate, tissue or organ, and Animalia, tissue, organ or organ system. Mode of nutrition as far as concerned. So Monera will show all types of mode of nutrition, autotrophic as well as heterotrophic. In autotrophic it is chemo and photos, and in hetero also we get to see lots of varieties, right? Talking about Protista, there is autotrophic and heterotrophic. Fungi is always heterotrophic. It can be sapro or parasitic and even symbiotic, right? And Plantae always autotrophic, while there are some which are parasitic, yes, and there are some which are partially heterotrophic. I'm talking about insectivorous plants. And Animalia, heterotrophic completely. Now, with this, let's move on to the next, and this picture. Figure 2.1, figure 2.1, guys, see here, just a second, I was just checking it. Yes, please. So this is the figure of different bacteria depending upon the shape. So the spherical ones are cocci, bacillus is rod shaped. So this is the spore formation. So spore formation is shown in bacillus here. It is shown in bacillus here. Oh wait, what have I done? Yes. Then spirillum, Treponema pallidum, that is an example of spirilla, and Vibrio is Vibrio cholera. Is that clear? Now, moving on to the next, we have this picture of eubacteria or cyanobacteria particularly. So in cyanobacteria, this is an example of nostoc, and we are able to see these special cells called heterocysts, which are specialized in nitrogen fixation because of the presence of enzyme called nitrogenase. Only those cyanobacteria can fix nitrogen which have these heterocysts, right? Then there is a mucilaginous or gelatinous sheath around it, which makes it slippery, So it is written gelatinous sheath, which makes it slippery. So the body, as we can see, is colonial and filamentous. This is the example of nostoc, blue-green algae involved in nitrogen fixation, right? Moving on to the next, this is figure 2.3, dividing bacteria. So this is fission being shown here, reproduces mainly by fission. See, this is the fission here going on. Is that clear? Moving on to the next diagram, that is of Protista. So the first diagram represents goniolux. Do you see this longitudinal flagella? And there is another flagella going through this transverse groove, right? Goniolux causes red tides, and we also get to see these cellulosic plates. Are we able to see the cellulosic plates? Yes or no? Right? So body is covered by stiff cellulosic plates which are present here. Yes or no? Everybody please let me know, are we able to revise things? So this is euglena. This is the example of euglena, which shows two flagella. There are two flagella written here. There are two flagella, one short, one long. So this is the long one, this is the short flagella, right? And this is euglena. Now euglena is a connecting link between plants and animals. It is a connecting link between plants and animals. So it shows mixotrophic. See, in the presence of light it will be photosynthetic, in the absence of light it will be heterotrophic, right? And the pigments are present exactly that of higher plants. They are identical to those of higher plants. Next diagram that we see here is that of slime mold. It is a saprophytic protist. So connecting link between this fungi and plantae, right? So it has characteristics of both fungi as well as plant like, right? So see, in favorable conditions it forms plasmodium. So the figure shown here is basically of plasmodium. This is plasmodium. Is that clear? This is plasmodium, right? And then in the unfavorable conditions it is going to form spores which will have true cell walls. Are these points clear? Now last diagram that you are able to see here is that of paramecium, the slipper organism, right? This is the contractile vacuole that is shown, contractile vacuole. Clear to all? And this is a ciliated protozoan. This is a ciliated protozoan. Is that clear? Let's move on to the next diagram now. The next diagram is that of, so these are the next diagrams. So the first diagram here, this represents Aspergillus. What is this? This is, this is mucor, Just a second, guys. Yeah, this is mucor. Wait a second. Yeah. So this one is mucor. This one is aspergillus. So will you be able to identify it when it comes for the identification? This first one is mucor, second one is aspergillus, and the third one is mushroom. Third one is mushroom. So mucor belongs to which category? Pram, that is Phycomycetes. And then aspergillus belongs to which class? Quickly let me know. Yes, aspergillus belongs to what? Aspergillus belongs to Ascomycetes, right? And mushroom belongs to, yes, please. Quickly let me know. Mushroom belongs to which category? You have to let me know, guys, in the chat. Let's quickly revise. Mushroom belongs to Basidiomycetes. Mushroom belongs to Basidiomycetes. Is this clear? Now, moving on to the next. So with this, guys, the diagrams of this chapter are almost over except for this one diagram. So this diagram represents that of TMV. This is a TMV. Clear? This is a diagram of TMV. This was the first virus to be discovered by Dmitry Ivansky, right? And what is the genetic material of TMV? It is RNA. And what is this? This is the outer protein coat. The protein coat is known as capsid, and each unit of capsid is known as capsomeres. Each one is known as capsomeres, right? Now this is a structure of bacteriophage. That means a virus which infects the bacteria. See the parts. First is head, second is collar, and third is these. So this is the sheath and these are the tail fibers. So you have to remember all these structures. Just a second, guys. All right, you have to remember all these structures. So head which will contain the outer capsid, and inside it will be present the genetic material. This is the collar, head, and the tail fibers. Are we clear, everyone? Please let me know. Is that clear? So with this, the second chapter is also done. Now coming on to the diagrams of chapter number 3, that is plant kingdom. Now watch carefully. First of all, let's see the diagram here. So guys, this diagram represents the volvox. Now I will strongly suggest you to write down the names here. So this is volvox, and what we see inside are the daughter colonies. The bigger one is the parent colony. So volvox is the colonial form. Volvox is a colonial form. We know that, right? And then this is Eulothrix. This is Eulothrix. Eulothrix is the filamentous form. It is a filamentous form of algae. Both represent green algae. Both are the green algae or chlorophyceae. Is that clear to all? Both represent green algae. Now moving on to the next. So these three diagrams, these all represent the brown algae or phaeophyceae. Now how do you remember the names? See, I have a trick. This long structure, this is laminaria. This is laminaria. Then the one which is dissected, this is fucus, which is dissected into two parts. Each will have two dissections. Each one will have two dissections. This is dictyota. This is called dictyota. So one laminaria, the long one, the only single stripe like structure, and then which is dissected further, that is fucus, which is highly dissected, and in fact every dissection is divided into two parts, that is called dictyota. Is that clear? So this is a way to remember. Now see, in brown algae, the parts are, what are the parts? The thallus is divided into holdfast, stipe, and frond. Holdfast is for attachment, stipe is a stem-like structure, not stem. Frond is a leaf-like structure, not leaf. But this frond is a lamina. This is the, see, it is a broad part which is actually going to be photosynthetic. That is why we call it leaf-like. This is stem-like. Are we clear? Then also we see some air bladders. These air bladders will help it in floating. So they provide buoyancy. They provide buoyancy. Is that clear? Right? Will you take separate session for scientific? Yes, Aruna, I will take that. I will take that. All right, guys. Now moving on to red algae. So red algae represents the rhodophyceae, RR. So red algae is rhodophyceae, right? So what are the examples? P2, G2. So here we see all the P2. Porphyra, this is Porphyra, while the other one is Polysiphonia. This is quite, sorry, this is Polysiphonia. Wait, this is Polysiphonia. Is that clear to you? And it has very similar plant-like body, see branches and main axis, while Porphyra only has this front. Are we clear? Are we clear with this? Quickly revising everything. Now coming on to this very important table of kingdom Plantae. So divisions of algae and their main characteristics. So we have green, we have brown, and we have red algae. So what are the pigments here? See, all of them will have A, because A is a universal chlorophyll. Other than C, see the pattern B, C, D. You have to remember CPR. Remember them in the order CPR, that is chlorophyceae, phaeophyceae, rhodophyceae, then A will be present in all, and then B, C, D. Then in phaeophyceae, there are some carotenoids, the most important carotenoids and xanthophylls, right? The most important of them is fucoxanthin. Fucoxanthin. In rhodophyceae, we have main pigment called phycoerythrin, which gives it red color. Talking about the stored food, very important. See, mannitol and laminaria, they are in phaeophyceae, while rhodophyceae will have Floridian starch. Chlorophyceae will be typical like a plant, just starch. Talking about the cell wall, see all the highlighted ones, they are the PYQs. Now talking about the cell wall, in chlorophyceae we just have cellulose and pectin. Cellulose and pectin, while in phaeophyceae we have cellulose and elgin, and in rhodophyceae cellulose plus pectin and polysulfate esters. Are we clear till here? Yes, Yuvan, what happened? I will be talking about scientists in a separate session, don't worry. I'm not ignoring anyone. But if I keep reading all the chat, who will study? Inert crystalline structure. Inert crystalline structure. Inert means it doesn't show any response, right? It will not harm. It is inactive when it is outside the living being. That is being inert. And crystalline means it can be, we can form crystals, right? All right, guys, moving on to the flagella. So both chlorophyceae, phaeophyceae are flagellated. In chloro, there are 2 to 8 equal flagella, and that too on the tip. So it is apical, just like this. On the apex, we'll have 2 equal flagella. While in case of phaeophyceae, the flagella are on the lateral side. One is long, another is short. They are unequal and they are lateral. Is that clear? While in rhodophyceae, both the flagella are absent. Now see, the habitat, chlorophyceae is freshwater, brackish is found in brackish as well as salt water. Phaeophyceae and rhodophyceae, they are rarely found in fresh water, right? Mainly in the salt water or brackish water. Are we clear? Now, moving on to the next, this diagram of bryophyta. So the first one represents the liverwort. First one is the liverwort, and which liverwort has been drawn here? This is Markentia. Now remember, Markentia is unisexual. It is unisexual. Call it dioecious. Why? Because the male-female thallus is different. What you see here in the diagram A, this is the female thallus. This is the female thallus, while B represents the male thallus. Is that clear? On the lower side, on the ventral side, we have, remember, on the ventral side, we have rhizoids. These rhizoids are root-like structures. They are not meant for absorption. They are only meant for attachment. Is that clear? Then, gamete cup present on both male-female thallus. They are the asexual buds, which will give rise to the respective thallus, right? And this is archegoniophore, while this is archegonia. Archegonia is the female sex organ. Female sex organ. Is that clear? Which will produce egg. While Entheridio4 is a stalk, just like Archegonia4, and on top of it we have Entheridium. Entheridium is the male sex organ giving rise to entherozoids. Are we clear till here? Yes or no? Please tell me. Are we clear? Now, moving on to the next diagram, that is a moss. So, this represents funaria. This one is of funaria, while the D diagram is a sphagnum or the peat moss. So, peat moss also has water holding capacity. That is why we use it for transshipments, right? So, peat moss, as you can see, it has both archegonial branch and entheridial branch. So that means it is bisexual. Can we say that or not? Yes or no? So it is bisexual. While in this funaria ka diagram, we are able to see rhizoids and main axis and leaf-like structures. So this represents the gametophyte. This represents a gametophyte. See, we know the gametophyte is also thalloid, but it is somewhat differentiated into root-like structures. This is root-like for attachment. This is stem-like structure, and there are leaf-like structures. Leaf-like structures are present in the spiral arrangement. So leaves are spirally arranged. Yes or no? So in both the cases, leaves are spirally arranged. And then sporophyte is attached to the gametophyte. Yes, sporophyte is attached to the gametophyte. Why? For food, for nutrition. So it is partially dependent on the gametophyte. Please tell me that these points are clear to all. Yes, guys. So in the sporophyte, there are three parts. There is foot, setae, and capsule. Foot, one. Then two is setae, and third is capsule. Capsule will have spore mother cells, which will give rise to spores by meiosis. Please tell me that these points are clear. Now, what is the dominant type? Sorry, dominant stage of gametophyte? That is gametophyte. Sorry, dominant stage in bryophytes is gametophyte. Please tell me these points are clear to all. Are we able to revise the diagrams and the topic along with it? Yes or no? Right? So I'll be sharing all these on the Telegram channel also. By the way, if anyone of you has not joined the Telegram as yet, it is Tina Ma'am Biology. I'll be putting up all these NCERTs on the Telegram channel. Yes, guys. Moving on to the next diagram, that is of pteridophytes. This first pteridophyte shown here. This first pteridophyte is Slegionella. This one is Slegionella. Sanjeevani Booty, Slegionella. Is that clear? So creeping body can we all see here? This is Slegionella. Slegionella is heterosporous, right? Slegionella is heterosporous. This is equisetum. This is equisetum. Now we know that the micro and the megasporophylls, in pteridophytes, most of them are homosporous, but in some of them they can be heterosporous. So there will be micro and megasporophylls separately. Now Slegionella and Salvinia, they are, see, this is Legionella and this represents Salvinia. These are both heterosporous. Salvinia, yes or no? These are both heterosporous, right? Now this represents the fern. This represents the fern, which is used for ornamental purpose. Now, in this diagram of equisetum, guys, are we able to see cone? So in few pteridophytes, these sporophylls, they will aggregate to form cone or strobilus. So cone and strobilus can only be seen in few pteridophytes. One example is equisetum. Second example is Legionella. It forms a cone as well. Cone is present in Legionella. Are we clear? Ma'am, Telegram is going to be banned. No, Haswant, not completely. There are some restrictions, but not completely. Don't worry about that. There are some restrictions though. I still have to explore the options yet. Let me tell you. All right. Now, this was all about pteridophytes. Will you be able to identify the diagrams? Please let me know. Will you be able to identify the diagrams, guys? Moving on to the next, that is gymnosperm. The first diagram represents the cycas. Now, what do we know about cycas? It is unbranched. It is unbranched. The cycas doesn't show branching, and moreover, it is unisexual. The male and female cycas are different. There are different cones present on this. And then this is pinus, while pinus is branched and it is bisexual. So both male and female cones will be present on the same plant. Are we clear? Yes, please. Are we clear here? And this diagram, last diagram, this represents ginkgo. This represents ginkgo. So you see a dwarf shoot, a long shoot, and these seeds. And we know that in gymnosperms, guys, there are naked seeds. In May 3rd exam, NEET 2026, there was just one question asked from this chapter, and that was related to gymnosperms only. Yes or no? This was around gymnosperms. So gymnosperms have naked seeds. See, the seeds are not covered. Yes or no? Please let me know these points are clear. The diagrams are done, and these two diagrams, one is of dicot and the second one is of monocot. How do we identify? Multiple factors. First of all, do we see that in this we have an isobilateral leaf, while in dicots we have dorsiventral leaves. So easily identifiable. Yes, please let me know, guys. So this is dicot and monocot, and with this then plant kingdom is also done. Moving on to animal kingdom. Yes, please give me a thumbs up that we are able to revise the diagrams and the tables here. Yes, please. Quickly let me know. Are the 3 chapters well done? So this is chapter number 4, that is animal kingdom. Moving on to the diagram, first diagram of the chapter. This diagram represents the radial symmetry. This diagram represents what? Radial symmetry. So what is radial symmetry? You can divide the organism into 2 equal halves in any plane. 2 equal halves in any plane. Is that clear? In any plane. Let me know in the chat which phyla have radial symmetry, guys. There are 3 you have to remember. One is Coelenterata, then we have Ctenophora and Echinodermata. Only adults have radial symmetry. Are we able to revise these things? Yes? Alright. Now the second diagram represents a bilateral symmetry. So bilateral symmetry is seen platyhelminthes onwards. Platyhelminthes till chordata, except in Echinodermata. But Echinodermata larva will also show bilateral symmetry. Are we clear here? Yes. The next diagram, see, the next diagram shows the number of germinal layers. So those with 2 germ layers, only ectoderm and endoderm, they are known as diploblastic. So this is the diploblastic, and the one with 3 germ layers, so ectoderm, endoderm, and the third one is mesoderm. This is a triploblastic. This is the triploblastic. Now in diploblastic, between ectoderm and endoderm, what do we have? We have mesoglea. We have mesoglea, which is non-cellular, hence non-living. Please tell me these points are clear to all. Right? So this was diploblastic and triploblastic. Which all organisms are diploblastic? Porifera, Coelenterata and Ctenophora. They are diploblastic, while platyhelminthes onwards till chordata we have triploblasts. We are revising the general features as well, guys. Now talking about the coelom. Now see, the first diagram shows the coelom. So this actually is a true coelom. Why? It is lined by mesoderm on both the sides. So see, this and this. So this pink layer, this also represents mesoderm, and this pink layer also represents mesoderm. So this is mesoderm, outer and inner. When the body cavity is lined by mesoderm on both sides, that is lined by mesoderm on both sides, this is called true coelom. So which all are the true coelomates, guys? True coelomate starts from annelida till chordata, that is true coelomates. Is that clear? Then where we see the scattered pouches, so these are the scattered pouches of mesoderm. So this is a characteristic feature only of Aschelminthes. They are pseudocoelomates. So such coelom is known as pseudocoelom. And here we don't see any body cavity. So these are known as acoelomates. Which one are the acoelomates? The first four phylum, that is Porifera, Coelenterata, Ctenophora, and even the first triploblast, that is Platyhelminths. Please let me know that these points are clear. Yes, guys? All right, moving on to the next. Now, this table can solve so many questions. So, see, these are all PYQs. So, cellular level of organization is only seen in Porifera, guys. Now, radial symmetry is seen in Coelenterata and Ctenophora, but those with acoelom and also echinodermata, by the way, adult. But it is acoelomates, hence radial and acoelomata. There are two phyla. Then bilateral symmetry will show three different types of coelom. Bilateral and acoelomate, answer is Platyhelminthes. Bilateral but pseudocoelomate, answer is Aschelminthes. See, whenever you hear the word cellular, you don't have to read anything, answer is Porifera. The cellular level is only shown by Porifera. Similarly, you hear the word pseudocoelomate, you don't have to read anything. Answer is Aschelminthes, right? While there are bilateral with true coelom, also known as coelomates. So, Annelida till chordata. Are we clear? Yes. See the first diagram of Porifera. So, the first diagram represents a very simple one, that is Sycon or Scypha. This second one is Euspongia. What is the general name for Euspongia? It is bath sponge. And this one is Spongilla or the freshwater sponge. What kind of symmetry are you able to see here? It is asymmetric, right? And do you see all these ostia, the pores over the body? Yes or no? Is that clear to everyone? Right? All right. Now, moving on to the next diagram. So, they can give you a diagram and ask you certain characteristics. So, you should be able to identify the diagrams, guys. So, the first diagram is that of Aurelia. This is Aurelia, right? And it represents which body form? It represents Medusa, the umbrella-like body form. While the second diagram is that of sea anemone or Adempsia. Adempsia or the sea anemone. And what type of body type is being represented here? That is polyp, plant-like body. These are the tentacles. These are the tentacles. And on these tentacles and the body surface, they have characteristic cells called stinging cells. Is it clear? So this is umbrella-like Medusa. Umbrella-like Medusa. Is that clear to all? So this is Coelenterata. Now the characteristic cells of Coelenterata or Cnidaria, the nidoblast, the stinging cells, guys, they are here. These are the stinging cells. See, these are the stinging nidocytes or the stinging cells. What are the functions of stinging cells? Very important. One is anchorage, second is defense, third is capture of the prey. So this is the toxin containing sac-like structure called nematocyst. This is the nematocyst, also known as stinging capsule. So this contains hypnotoxin. This contains hypnotoxin, and this is the thread tube. Please tell me these points are clear. Yes, guys. Are we clear? Right. Moving on to the next diagram, which represents a ctenophora or pleurobranchia. So these green things, these are 8 ciliated comb plates. These are the 8 comb plates. While these are the tentacles, which have what? Coloblasts, right? And it also shows bioluminescence. So do we see 8 comb plates? 1, 2, 3, 4, 5, 6, 7, 8. 8 comb plates is a characteristic feature of ctenophora, right? Now moving on to the diagrams of Platyhelminthes. The first one represents the tapeworm, also known as the taenia, right? Both are flatworms, while the other one is liver fluke. Is that clear? So liver fluke causes also known as fasciola. This is also known as fasciola, going to cause liver rot or fasciolopsis, and while taenia is going to cause teniasis. Is that clear? Both infections occur by contaminated food and water. Now, this diagram represents the male and female of Ascaris or any roundworm. This is Ascaris, the common roundworm. Do you see the female is long and slender, and it has a straight posterior end? Do we see all these structures? While the male Male has a curved posterior end. Male has a curved posterior end. Is that clear? Curved posterior end in case of male, and also it will have penile setae, while the male is short and broad. These are the features of sexual dimorphism. So we see the sexual dimorphism for the first time in case of Aschelminthes. Hi Shani. Now, moving on to the next phylum, that is Annelida. Annelida word comes from rings. So, are we able to see the rings present on the body? So, rings are known as annulus. These rings are going to divide the body into different segments. All these segments are known as metameres. So, Annelida represents the true segmentation, metameric segmentation here. Is a clear segments known as metameres, right? The first one is the structure of Nereus, while the other one is leech, and leech has the sucker to get attached to the host body. Leech is ectoparasite. It stays outside the body and sucks blood. The scientific name is Hirudinaria, blood-sucking leech. It is sanguivorous, blood-sucking, sanguivorous. Now, this is parasite, while Nereus is totally see, Nereus is aquatic as well as it is dioecious, means it is unisexual. So male female Nereus are different. The structures that you are able to see here, these are parapodia. These are parapodia. Please tell me these points are clear to all. Yes guys, please tell me these points are clear to everyone. Yes, so these are the structures. This is Nereus. Nereus is aquatic, by the way. That is why these parapodia are going to help in swimming. So Nereus is aquatic. The third example of Annelida is earthworm, and I am sure we all remember and recognize the structure of earthworm. Now moving on to the largest phylum of animal kingdom, that is Arthropoda. Look at these structures. The first structure is that of locust. Locust is a gregarious one. Written or not, it is a gregarious pest. That means it moves in large groups. This is your butterfly, insecta. Then this is the scorpion, which is viviparous. It is viviparous. It directly gives birth to the young ones. And the last one is that of prawn or the paleomon. Is a paleomon. Is that clear? Look at the other examples also. The other examples we know, one is Apis, Bombyx, Lacifer. Vectors are Anopheles, Culex, Aedes, and living fossil is king crab. Yes or no? So there are some examples which are given in this format, economically important, and while other examples are given in the form of diagram. Next guys, we have the structure of Pila. Pila is the apple snail. It is the apple snail. These are the examples of mollusca, second largest phylum. While this is the devilfish. Devilfish is octopus, the master of camouflage. Octopus. Is that clear? The outer side we see a calcareous shell made up of calcium carbonate, and the soft body is present within that. Is that clear to all? All right. Now moving on to the next diagram, which represents the echinodermata. The first one is Asterias, which is a starfish. Asterias, that is a starfish. And do we see it? It will have radial symmetry. Yes or no? Radially symmetrical adults. While the larva not shown here, but they will have bilateral symmetry. And this one is Ophiura, and Ophiura is the brittle star. See guys, once and for all, for last time guys, we are going to revise these diagrams. So please don't get your energy droop at any point of time, and I would strongly suggest for the last time you have to write down all these names with your own hand right beside the diagram. I know they are written beneath it, but write it beside it. One, you are going to practice it with your own hands, and it will be the final revision. Is that clear? And be very careful with all the diagrams. Now talking about this Belanoglossus, which represents the phylum Hemichordata. Hemichordata. So what are the three parts of Hemichordata body? Proboscis, this is collar, and this one is trunk. Rectify that. Is that clear? And this structure represents the gills, and proboscis will have the proboscis gland. It will have a Proboscis gland. Proboscis gland is going to be excretory in nature. Is that clear? So it has proboscis, collar and a long trunk. Is that clear to all? This is melanoglossus, looks like tongue, so we also call both of them as tongue worms. We call them as tongue worms. Is that clear? Worm-like body. Now, moving on, figure 4.16. This is a general structure of a chordate. What are the features which a chordate must possess? One is a nerve cord, but it has to be dorsal. The dorsal hollow nerve cord is a feature of chordate. While in non-chordates, it is ventral and it is solid and it is double. While here it is single. Is that clear? Then talking about notochord, the second feature, notochord is again a rod-like structure, rod-like, which is solid, again dorsal. And this notochord may or may not persist, but it should be present at one point of time to authenticate some organism as a chordate. Without notochord, nobody can be called as a chordate. Is that clear? Now, nerve cord will give rise to central nervous system eventually. Notochord, if it persists, may or may not get modified. If it gets modified, like in our case, it gets modified into vertebral column. That is why we are known as vertebrates. Then there is a post-anal tail, and fourth is the gill slits in the pharynx area, so pharyngeal gill slits. Is that clear? These are the four common features of chordates. Now talking about the comparison, see notochord present, here it is absent. Central nervous system is dorsal, hollow and single. What are we talking about? We are talking about the nerve cord just discussed. While here it is ventral, solid and double. Pharynx will have gill slits will be absent here. Heart is always ventral, and in case of non-chordates, if it is present, it is dorsal. Post-anal tail is present here, it is absent in here. Are we clear? Look at this diagram, guys. This represents your urochordata, that is ascidia. Remember, urochordata will have siphons. So these are some siphons through which water moves in and water goes out. So these are the inlets and outlets of the siphon. Is that clear? Now, let's see this table. So phylum Vertebrata is further. So this is subphylum Vertebrata. Okay, so this is further divided into two divisions based on what is the basis of classification here. Further classification of vertebrata is on the basis of jaw. This is on the basis of jaws. Those which have jaws are known as gnethostoma. Those without jaws are known as agnetha. Is that clear? We have one class under it, cyclostoma. While in gnethostoma, there are two super classes based on their locomotory structures. Those which have four limbs, they are called tetrapoda, while those which have fins, they will be known as pices. So chondrichthyes and osteichthyes based on their type of skeleton, internal endoskeleton. If endoskeleton is made up of cartilage, it is called chondrichthyes, while if it is made up of bones, it is known as osteichthyes. Are we clear everywhere? Everyone, please let me know. Make a WhatsApp group. Okay, Meena, we'll talk about that. Yes, guys, are we clear here? All the serious students, take a minute. Take a minute and let me know. Are we clear here? I'm talking about all the serious students here. Yes? Perfect. All right. Let's move on now. So, tetrapoda can further be divided into four classes, that is amphibia, reptilia, aves and mammals. This is the diagram of a jawless vertebrate. We are talking about cyclostoma, jawless vertebrate, that is cyclostoma. Example is petromyzon or lamprey. These all highlighted lines are PYQs. So see, cyclostoma means it has a circular mouth. It has a sucking circular mouth without jaws. These are the features of cyclostomata. And are we able to see these gill slits? So it has 6 to 15 gill slits. Yes or no? It has 6 to 15 gill slits. Are we able to see that? Now, moving on to the next diagram, which is that of chondrichthyes, cartilaginous fishes. The first one is scoliodon. First one is scoliodon, and the second one is pristis. Pristis is a sawfish. Do you see the mouth here? So the mouth has a saw in front of it, hence sawfish. Is that clear? And scoliodon is the dogfish. Next diagram represents the bony fishes. The first one is hippocampus. Hippocampus is a sea horse. It is a seahorse, also known as hippocampus, right? While this one is catla catla. Clear to all? Now, moving on to the next. Now, this might look like a lizard, but please remember this is not a lizard. It is a salamander. It is a salamander. And this one is frog, rana tigrina. Are we clear? Right. Now, moving on to the next set of diagrams. This represents all the reptiles, guys. By the way, this diagram of lizard is not even given in your book. So just remember, a diagram which looks like lizard is not a reptile. It is not a lizard. It is a salamander, which is an amphibian. Are we clear? Can we remember it like that? Yes. So these are all reptiles. The first one is chameleon. First one is chameleon, the master of camouflage. Yes. Then it has a sticky tongue like frog to take in the insects. This is crocodile. One exception in class Reptilia with 4-chambered heart, while all others will have 3-chambered heart, right? This is tortoise. Sorry, this is turtle. Wait. This is turtle or chelone. Tortoise is testudo. This is turtle. Look at the limbs. So, turtle is mostly aquatic, so it has these structures which help in swimming. And this is snake called Naja Naja. Is that clear? Right? And Naja is a poisonous snake. Remember, what are the 3 examples of poisonous snakes given in here? Naja is cobra. This is cobra. It is poisonous. Bangaras, that is crate. And vipera, that is viper. Clear to all? Now, another set of diagrams which represent all the birds. The first one is neophron. Neophron is vulture. Neophron is vulture, right? It is a scavenger, guys. It is a scavenger. Then this one represents ostrich. Are you writing all the examples with me on your book? Please let me know. Yes, guys. Are you writing down the examples with me? Everyone? All the serious students here, are you writing the names along with me? Yes, Vishalini is doing that. Very good. SP, this is an English batch, bache. I try to keep it very simple. Are you all writing with me? Yes? Good. So this is ostrich. Ostrich is also known as struthio. It is also known as struthio. This is peacock. Sorry, what is it? This is parrot. I was looking at peacock and writing on parrot. And this is a peacock, the national bird. So peacock is pavo, and this is parrot, which is psittacula. This is psittacula. Are we clear? Yes? All right, guys. And by the way, ostrich, struthio, this is a flightless bird. Yes or no? This is a flightless bird. All right. Now, coming on to the class Mammalia. So, the first diagram is that of Ornithorhynchus. Ornithorhynchus is platypus, it is platypus, duck-billed platypus, and it is oviparous mammal, egg-laying mammal. Right? This is pteropus or kangaroo. Marsupial, pouched mammal. Marsupial means they have pouch. See, do you see the young one in the pouch? So marsupial or the pouched mammals. While This one is Pteropus. Pteropus is a bat. And this one is Lanothera or the blue whale. Both of them, they are placental mammals. This and this are placental mammals. Is that clear to everyone? Right. Now, so this is the table which summarizes the entire animal kingdom and we have discussed it already. Moving on to the next chapter, guys. This is morphology of flowering plants. Everybody on chapter number 5? Give me a thumbs up. Yes, guys. Are we on the chapter 5? Have we written everything previously? Ma'am, kangaroo is a macropus. What have I written then? Yes, macropus. Wait. What did I do? Did I write something else? Yes, kangaroo is macropus. Okay, by mistake, I think I've written something else, but this is kangaroo, guys, and this kangaroo is macropus. Yes, it is a pouched mammal. It is a pouched mammal. Is that clear? All right. All right, guys. Moving on now to the next, that is morphology. Chapter number 5, let's see the diagrams of morpho. The first diagram represents the different parts of a flowering plant. So, yes, the overall plant can be divided into two parts. One is the root system and second is the shoot system. Now, this diagram represents a tap root system where we have a primary root and secondary root. So, this is a tap root system. This is a tap root. While the aerial part is the shoot system which has flowers, and do you see the fruits and the stem and the leaf and the a region from where any part develops, that is called node, and that space between the two nodes is internode. Are we clear? So this is this diagram represents the parts of the flowering plant. Now we should be able to differentiate these diagrams, these different type of roots. This is a tap root. This is a tap root where we have a main root called the primary root, and then we have the lateral, secondary or tertiary, and so on. Then this is a fibrous root. See, tap root develops directly from radicle. Yes, while fibrous root, in fibrous root, the radicle gives rise to the primary root, but primary root is very short lived. Yes or no? So later on, so the primary root degenerates, and later on these fibrous roots originate from the base of the stem, originate from base of the stem. Yes or no? They originate from the base of the stem. And the third one is adventitious roots. Adventitious roots arise from part. They arise from part other than radicle. Other than radicle. Usually supportive in nature. Are we clear? Yes. Moving on. So the next diagram is that of regions of the root, guys. Figure 5.3. Regions of the root. First of all, we have a root cap which protects the meristematic zone. So very distal to the plant, we have region of meristem, right, where active cell division occurs. The cells are small. This is the region of meristem. Then proximal to that, we have region of elongation, which is actually responsible for growth, because of increased vacuolation and enlargement of the cell. And third is the zone of maturation or root hair zone. This is also known as root hair zone. Of course, the name suggests that it will have root hairs, which will increase the surface area for water absorption. Yes. Now, structure of leaf, guys. This is a typical structure of the leaf. So what are the three parts of the leaf? Number one, leaf has a leaf base. Second is the stalk known as petiole. Third is the leaf blade called lamina. Yes. So leaf base is that part with which it is attached to the plant, right? And now leaf base can show various modifications. For example, it can have two small leaf-like structures called stipule. Yes, it can have stipule. See, two lateral small leaf-like structures called stipules. Yes, in case of monocots, it will form a leaf sheath. It will form a sheath, right? While in legumes, guys, one, two, and wait. So this is one, this is two in monocots, and in legumes, guys, it is swollen, leaf base is swollen to form pulvinus. Is that clear to all? While petiole may or may not be present, and this is the lamina, where we see a network of veins and veinlets, right? Now, in the diagram B and C, we are able to see the venation. So this is a reticulate venation where a network of veins and veinlets is formed. Reticulate is a characteristic feature of dicots, but is it present in all dicots? No, right? And similarly, this is the diagram representing the parallel venation, which is a characteristic of a monocot. Do we see all the veins? They are running parallel to the midrib. Is that clear to all? This is the midrib. This is the midrib. Are we clear? Now, moving on to the next diagram, which represent types of leaves. So, both the diagrams given in here represent the compound leaves. What do they represent? Compound leaves. See, how do we identify whether it is a compound leaf or a simple leaf? So, compound leaf, a bud. See, a bud is present in the axil of petiole in both simple, not in the axil of leaflets. So what we see here are all leaflets. These are all leaflets. And we don't see any bud in its axil, while there is a bud here. So everything present ahead of that is a compound leaf, and compound leaf has leaflets. So this one is pinnately compound. This one is pinnately compound. In pinnately compound, guys, there is this midrib, which becomes, midrib is known as rachis, and the leaflets are arranged around it. While in palmately compound, the example is silk cotton. This is neem. Pinnately compound is, example is neem, and palmately is silk cotton. All the leaflets are arranged at the tip of the petiole. This is the tip of petiole. Yes or no? And there is a bud here in the axil, not in the axil of the leaflets. Are we clear? Please let me know. So the diagram and examples are important. While now we see figure 5.6 showing you phyllotaxy. Phyllotaxy is the arrangement. See, phyllotaxy is the arrangement of leaves on the branch of the stem, right? Now, China rose is an example of alternate phyllotaxy. There are other examples also. China, mustard, and sunflower, they all represent alternate phyllotaxy. So, one leaf per node. Do you see at every node there is one leaf only? Yes. While in guava we have opposite phyllotaxy. So, one node, two leaves, one node, two leaves, one node, two leaves. This is opposite phyllotaxy. Another example of opposite is calotropis. Then there is world. In case of world, guys, there are multiple leaves around the same node. Yes, everyone? So, example is Elstonia. Please tell me more than two leaves at a node. This is world. Example is Elstonia. Clear? Now, this diagram shows the racemose inflorescence. What is racemose? In racemose, the main axis continues to grow. Do we see the main axis continues to grow? Here, main axis continues to grow, right? So, the new flowers will arise. The new flowers, these are the new ones, new flowers at top. So what do we call this as? This is known as acropetal succession. This is called acropetal succession. So racemose inflorescence will always have acropetal succession. Yes or no? It will have acropetal succession. While in case of cymose, guys, see, the tip will terminate into flower. Tip terminates into flower, and the new flowers will arise from the base. So the tip which was supposed to continue to grow, that terminates into the flower, and the new flowers will grow from the base, new at the base. So this is known as basipetal. This is known as basipetal. When the new is at the base, this is called basipetal. This is a feature of cymose inflorescence. Are we clear? Very important diagram, already a PYQ. So this diagram has already come up. So they have asked what type of flower do these diagrams represent. So see, I think it was a question in 24 or 23 perhaps. So both they have given this B and C diagrams and asked for the same. Now the first diagram represents the hypogynous flower. Both B and C, they represent the perigynous condition, while D represents the epigynous condition, right? So see, how do we identify? So in hypogynous, hypogynous means below the ovary. So ovary is superior, superior ovary. All the structures are arising. So this is the superior ovary. Everything else is arising below the ovary. That is why hypogynous. Now talking about perigynous, so this might not, come true to the definition actually, because in perigynous everything is present at the periphery at the same level. So this B diagram actually is confusing, but you just remember what is given in NCERT. In perigynous, ovary, all the other floral structures, they are at the same level, right? While in case of epigynous, epigynous we have inferior ovary. This is inferior ovary. Everything else, all the other floral structures, they arise above the ovary. Do we see? They are arising above the ovary. This is inferior ovary. While in perigynous, we have half inferior ovary. The diagrams are important, examples are important. And by the way, guys, I'm going to share all the examples, all the cheat sheets today. All the cheat sheets. Is that clear? All right. So learn the examples very well. Now parts of a flower. So look at the diagram, how they have represented it. This is androecium, and look at the way they have shown gynoecium. It may confuse you, yes or no? Then this is corolla, that is the aggregate term for all the petals, and calyx, and this is the stalk of the flower known as pedicel. Look at the calyx. Corolla is easily identifiable, then androecium is identifiable, but look at the diagram of gynoecium. Doesn't look like a typical gynoecium, yes? But just remember how they have shown it. Now this is estivation. So margin to margin, this is valvate. No overlapping at all, just margin to margin, no overlapping. This is valvate, guys. Second is twisted. In twisted, one margin is out, other margin is in. Similarly, out and in, there is a regular pattern. This is called twisted. This is called twisted. So margin overlaps the next one. This is twisted. Then this is imbricate, in which there is no fixed pattern. No fixed pattern, right? Both the margins are inner side. One is outer, one is inner. Both are outer. Are you getting it? So no fixed pattern. No fixed pattern. While the last one, this is a characteristic feature of Fabaceae. This is a characteristic feature of Fabaceae, where we have vexillary. This is vexillary, also known as Papilionaceous, because found in Papilionaceae, which is a subfamily of Fabaceae only. Papilionaceous. So in Papilionaceous, guys, the characteristic thing is there are always 5 petals. There are always 5 petals. The larger one is known as standard. These ones are laterals, and the smallest ones are known as keel. How many keel? 2, and they are fused. Lateral is again 2 but not fused. Standard is 1. Standard is towards the posterior side. This is towards the posterior side. Keel is towards the anterior side. Please tell me all these points are clear. Is that clear to everyone? Now, moving on to the placentation. Very important. Placentation. See, the first one represents the marginal placentation. This is marginal placentation as seen in pea and beans. So, placenta forms the ridge. So, this is placenta forming the ridge on which forming the 2 rows and on which these ovules are present. Now, Let's talk about this one. This is the exile placentation, where we do see this is the septum which is dividing the ovary into multi-locules. So this is multilocular, like you see in tomato. This is multilocular ovary, right? And this is a placenta. Clear? Now this is parietal. Parietal, present ovules all along the inner wall. It is one chambered. And for parietal, remember, there is false septum. Right? These are the key features. While this one is a free central. This one is free central. There is no septum at all. In free central, again, septa are absent and it is monolocular. Monolocular. No septum at all. When there is only single ovule at the base, which is called basal. This is called basal placentation. Please let me know all these diagrams are clear to you all. Yes, guys. Are all the diagrams clear? Are all the diagrams clear? Now, this diagram represents the structure of a fruit and not any other fruit. It is drupe. It is drupe. So see, what is the definition of a drupe? Drupe is always represented by monocarpellary condition. It is always monocarpellary with superior ovary and only one seed. Only one seed. Is that clear? Now we have two examples in this. One is mango, which is a dicot, and this is coconut, which is a monocot. Secondly, here the outer fruit wall is known as pericarp. Pericarp is divided into epi, meso and endo in such a way that epicarp is, see, outer and thin epicarp, middle fleshy or edible mesocarp, like in case of mango, and then coconut, sorry, in both the cases we have inner stony endocarp, right? In coconut, the mesocarp is fibrous. This one is mesocarp fibrous, while here mesocarp is fleshy and edible. Are we clear? Please let me know. Are you clear about these two? So drupe, the two examples are mango and coconut. Clear to all? Alright, now let's see the structure of a dicot seed. So of course it has a seed coat. There are two seed coats, testa and tegmen. Testa and tegmen. Yes, there are two cotyledons. Can we see? Cotyledons are also known as seed leaves. Also known as seed leaves. So upper structure represents the plumule, the lower one represents the radicle. This is the embryonal axis. Also in the seed, guys, we see a micropyle. What is micropyle? It is a small hole for absorption of water, for germination and for gaseous exchange. Gaseous exchange is important, right? Germination demands a lot of energy. And then this is the hilum. What does hilum represent? Hilum represents that part, it is a scar through which the developing seed is attached to the fruit. This is hilum. Are we clear? Are we clear here? Now let's see the structure of monocot seed. Look at this monocot seed. So the outer layer represents a seed coat and fruit wall, that is pericarp. They are fused together. They are fused and it is membranous. It is transparent. The inner region to that is the endosperm. Endosperm is nutritive. Endosperm is not completely consumed in monocots. That is why monocots are endospermic seeds. And outer to the endosperm, do we see a protein layer known as the eleuron layer? Everyone? Yes? Then beneath the endosperm, tilted on one side, we have one cotyledon, because this is monocot. So there will be one cotyledon known as scutellum, right? And this is the embryonal axis. This is embryonal axis. Upper part we have plumule, and the covering sheath of plumule is known as coleoptile. while on the lower side we have radicle, and the sheath of radicle is known as coleorhiza. These structures are absent in dicot. Yes guys, are we clear? Everybody, let's take a minute. Please let me know, are we clear here? Yes? All right. Moving on to the next, this is a typical floral diagram with floral formula. So this is a floral formula and this is the diagram. What does this dot indicate? Mother axis. The outer layer is that of sepals. Yes, that is calyx. The inner one represents the corolla, and inner to corolla we have these stamens called androecium, and this is the gynoecium. So first of all, from the diagram, can we see how many calyx are, how many sepals are present? Just count them. 1, 2, 3, 4. Is that clear? Are they fused? No. So we will not put a bracket. Yes. Now, see here in this 4, do you also notice that 2 are on the outer side, 2 are on the inner side. So instead of writing 4, we will write 2 plus 2. 2 plus 2. Clear? Then talking about the corolla, the petals, they are all in one line. They are not, and how many are they? 4. They are not fused. Are we able to see the estivation also? Please tell me, are we able to judge the estivation also from this floral diagram? Yes, so floral diagram will tell you about the estivation. How many are there? How is the arrangement? So estivation, whether the fusion has occurred or not, or means cohesion and adhesion, everything will be depicted through the floral diagram. Is that clear? Then inner to corolla we have androecium. Can you count the number of androecium? You have 1, 2, 3, 4, 5, 6. So total 6. But we don't write it as 6 here because 2 are on the outer side, 4 are on the inner side. So 2 plus 4. Yes or no? So we'll write it 2 plus 4. This is known as tetradynamous condition, right? And then we have G, gynoecium. So how many are there? 2. How many are there? Wait. So how many are there? 2. And since there is one circle outside it, it means they are both fused. So we'll put a bracket around it. Now, whether it is superior or inferior cannot be depicted through the, cannot be showed through the floral diagram. And then this is the formula. In formula, how do we write it? So formula tells you about, see, the floral formula. There are certain symbols. BR for the bracteate condition, K for the calyx, C for corolla, P for perianth in case petals and sepals are indistinct, A for androecium, G for gynoecium. G above the bar is superior, G below the bar is inferior. So this is a sign for the male, this is a sign for female. While when both the symbols are combined, it becomes a bisexual. Plus encircled is actinomorphic, that means radial symmetry. Examples are important, guys. While percentage sign represents the zygomorphic. Now bracket will show fusion. Bracket will show fusion, while adhesion is represented by the line drawn above the symbols. Is that clear? Please tell me all these points are clear. So this diagram is that of Solanum nigrum or Mako belonging to the family Solanaceae. Is that clear? And this is the floral diagram. You should be able to. Yes, guys. Is that clear? So this is the floral formula and the floral diagram. You should be able to identify. Clear to everyone? So with this, guys, we complete the chapter Plantae also. Talking about anatomy. Moving on to anatomy, guys, quickly. This diagram has already come up in the exams. This is a PYQ already. So this represents the stomatal apparatus. See. So this diagram, this bean-shaped cell is a guard cell. The pore in between is a somatal pore or simply known as stomata. Now these cells surrounding the guard cells, these are subsidiary cells. These are subsidiary cells. So subsidiary cells, guard cells and stomatal pore together form the stomatal apparatus. Now this is a structured, this is a guard cell of dicot. How do you identify? In dicot, the guard cells are bean shaped. They are bean shaped. Yes. Now secondly, also notice that the guard cells have outer wall is thin, while inner wall is thick. Is that clear to all? Inner wall is thick. Now the dumbbell shaped guard cells. This is a characteristic of monocot. In monocot, there are dumbbell shaped guard cells. Please tell me all these points are clear to all. Yes, guys. Are these points clear to everyone? Now, let's see the different types of vascular tissue. Now, here xylem and phloem are alternating with each other. Yes. So, when xylem and phloem are present in alternate manner and different radius. So, xylem and phloem alternate manner and different radius, right? So, this is a different radius. So this is radial type of vascular bundle. These are radial type of vascular bundles. So radial are always found in roots, be it monocot or dicot. Then when xylem and phloem are present in the same bundle, they are present jointly along the same radius, This represents conjoint. This is conjoint. Is that clear? The difference between the B and C diagram is, in xylem and phloem, there is no cambium in between. When there is no cambium, means no secondary growth, hence it is called closed type of conjoint vascular bundle. While here, because cambium is present and secondary growth will be occurring here, so we call it open type of vascular bundle. So it is the presence or the absence which will determine whether it is open or closed. Yes, please. Are we clear? Is that clear to everyone? Right? All right. Let's move on to the transverse section. This represents a transverse section of dicot root. So how are we going to identify whether it is a dicot root or a monocot root, guys? So the difference will be on the basis of the vascular bundles. Monocot root. See, in dicot root, we see the vascular bundles are 2 to 4 in number. While in monocot, the vascular bundles are more than 6. That is why we call this condition as polyarch condition. Yes. Also, the pith here, although if you compare the diagram, the pith of the dicot appears larger, but actually the pith is small and inconspicuous. Pith is small and inconspicuous in dicot, while in monocot, in case of monocot, the pith is large and well developed. The pith is large and well developed, and there are more than 6 vascular bundles. So this condition is known as polyarch. So you will be able to identify the diagram on the basis of vascular bundle. Just count the number here. See, it is 1, 2, 3, 4, 5, 6, 7, 8. More than 6. More than 6, it is 100% monocot. Rest, all the structures are similar. That is root hair. Root hair are present. Epidermis, also known as epiblemma. We have multi-layered cortex made up of parenchyma. Then endodermis, which has suberin deposition, and hence Casparian strips are formed. So pericycle, few layered thick walled parenchyma, protoxylem and metaxylem. Now in this case, if you look carefully here, the protoxylem is on the outer side. So how do you identify whether it is proto or meta? See, the elements of protoxylem are always smaller. See, elements of protoxylem are always smaller. So the smaller ones are on the outer side. So when the smaller ones are on the outer side, do you notice? The smaller ones are on the outer side. Proto is outer, hence it is exarch condition. So both dicot or monocot will have exarch condition. That is, root will always show exarch and radial condition for sure. It can be open, it can be closed. Dicot is open while monocot is closed. Are we clear? Yes or no? Rest, there are similarities that have already been mentioned. Next is the structure of dicot stem. See, again, will you be able to identify the diagram? Characteristic ring arrangement of vascular bundles. Ring arrangement of vascular bundles is a characteristic of dicot stem. Yes or no? So these are epidermal hair, also known as trichomes. Do we see them multicellular here? Are we able to? Epidermis, single layered, hypodermis made up of collenchyma, parenchyma, endodermis. Parenchyma is made up, parenchyma forms the cortical layers, and the innermost layer is endodermis, and endodermis here is also known as starch sheath because it has lot of starch grains. Pericycle, do we see here? Pericycle is semi-lunar patch. What is it made up of? It is made up of sclerenchyma. So are we able to appreciate that here the cortex layer, cortex is highly differentiated and well developed. So hypodermis, parenchyma and endodermis together they form the cortex. Yes or no? Right? Then pericycle, semilunar patches made up of sclerenchyma just above the phloem. Yes, this is phloem. And do we see these cells? These cells represent the cambium. Yes. And then how do you identify whether it is endarch or exarch? The smaller element represents what? Protoxylem. Proto on the inner side. So in stems, guys, we always have endarch condition. So whether it is exarch or endarch depends upon the position of protoxylem only. It depends upon the protoxylem only. Are we clear? Yes, guys. Everyone? So stem will always have endarch condition and conjoint vascular bundles. Why? Because both are present within the present on the same radius. Yes or no? So what type of vascular bundle it is? Conjoint, endarch, and because there is cambium, hence open vascular bundle. So while describing a vascular bundle, you always have to tell these three aspects: whether it is radial or it is conjoint, second endarch or exarch, or whether it is open or closed. Are we clear? Yes, please. Are we clear? All right. Alright guys, now moving on to the diagram of monocot stem. So see, in monocot stem, very easily identified by the help of vascular bundle. See, vascular bundle is the key to identify the transverse section, right? You see radial arrangement, it is 100% root. If it is between 2 to 4, it is dicot. If it is more than 6, it is monocot. Yes or no? Right? Then if it is a dicot stem, there has to be ring arrangement. While if the vascular bundles are scattered, like in this case, it is 100% monocot stem. Now on the the vascular bundles on the outer side, those on the outer side, outer ones are smaller, outer vascular bundles smaller, and the inner vascular bundles, that means towards the center, inner vascular bundles, central vascular bundles, they are larger, right? Now cortex is not well developed. You see epidermis, and just below that we have a very reduced, highly reduced hypodermis. Rest of the ground tissue is not differentiated, ground tissue undifferentiated, so we don't give them different terms like endodermis or pericycle or anything. We don't use these terms. So ground tissue is undifferentiated. Are we clear? Now see the detailed structure. Again the vascular bundle. Since xylem flowing in the same bundle, hence it is conjoined. Because it is monocot, it has to be closed. Monocots show closed vascular bundles. And then being a stem, it will always be end dark. Is that clear? Now there are some special water cavities being noticed here. Do you see these two eyes? These are the water cavities. Now these water cavities are formed by the degeneration of xylem parenchyma. Okay, and they are present only in the monocot stem, not in the dicot stem. Are we clear? Please tell me. And then, so there is no differentiated pith or so on. We just call it ground tissue. While in case of dicot stem, do you see this whole big structure is pith, is very well developed, very large in dicot stem. Also guys, in 2026, the question was asked around medullary rays. What are the medullary rays? This structure, these are the radially arranged parenchymatous cells. This is medullary rays. Is that clear? So this structure represents the medullary rays. See this, and it is made up of parenchyma. It was asked in 2026 already. Yes guys, are we clear? Are we clear? All right. Next diagram is that of dicot leaf. Look at the differences, the dicot leaf and the monocot leaf here. So in dicot leaf and monocot leaf, we can clearly see the distinction on the basis of structures present in upper and lower epidermis. So this is upper epidermis called addaxial and the lower epidermis known as abaxial, right? Similarly, upper here and lower here. Now, this one is the dorsiventral leaf. Dorsiventral leaf DD present in dicot. While this is isobilateral. Iso means equal, bi means two, lateral sides, equal two sides. This is in case of monocots. Now see the distinct difference between the two sides. In dorsiventral, we don't see any stoma on the addaxial side. There is no stoma or no stomata. Rather, we do see a cuticle here, but no stomata. While on the abaxial side, we see stoma. But in case of isobilateral, the presence of stomata on the upper and lower side is equal. Is that clear? You can call it stoma, you can call it stomata. It is similar. Cuticle is also equally distributed or distributed on both the sides. Is that clear? Now, talking about the ground tissue. Ground tissue is known as mesophyll here. So, in case of dorsiventral, mesophyll is differentiated into palisade mesophyll and spongy mesophyll. Why do we call it spongy? Because of these air cavities. Look at the arrangement of these spongy mesophyll cells. And while the palisade mesophyll present just beneath the below the upper epidermis, they are very compactly arranged, right? Now, it has maximum chloroplast. It has maximum chloroplast. So, maximum photosynthesis is going to occur in the palisade mesophyll. Are we clear? Please let me know. Yes, guys. Anyone with any doubt here? Right? Now, also we do see a substomatal cavity just above the stoma, whether it is isobilateral or dicot. So, this is a similarity here. While in case of isobilateral, there is no distinction in the mesophyll. It is not differentiated. So, we see similar type of mesophyll in the isobilateral. See, all are, they are rounded in shape. There is no distinction into palisade or spongy. Are we clear? And see the arrangement of the vascular tissue. So, this outer layer represents the vascular bundle. These cells are bundle sheath cells. Bundle sheath cells are always sclerenchymatous, and these cells will surround the vascular bundle, right? This is the bundle sheath cells, and inside we have xylem and phloem. And in the isobilateral leaf, see here, we have this xylem with water cavities, and this is the phloem on the lower side. So, xylem on the upper side, phloem on the lower side. Are we clear? Are we clear? So guys, with this we are able to complete the diagrams of anatomy. Yes, moving on to the next chapter. Let's take a second guys, all the serious students. Yes, please, all the serious students, quickly let me know. All these chapters are well done and there is no doubt. If there is any, please let me know in the chat. Crystal clear? Perfect. Nobody sends me green hearts now. Right? Alright. Moving on to, but I miss green hearts. Never mind. Alright. Let's talk about the structural organization in animals. So, see the diagram of frog here. Everyone. Why do I have to ask for it? ऐसे थोड़े होता है. This is not the way. You should send them without being asked for. Alright guys, let's talk about the morphology of frog. See, the body is divided into two parts. One is head and second is trunk. Yes. So the neck and tail are absent. This is something we already know. Head has mouth and the pair of nostrils. Eyes are bulged. So see, these are simple eyes and they are bulged. And also they are covered by nictitating membrane to protect them inside water, right? This is about eye. So this is the head. Head is again triangular, right? And this is the mouth. Mouth is ventral on the lower side, very large and ventral mouth. Now this represents another eye and this represents the tympanum. This is the tympanum. Tympanum represents the outer ear. Yes or no? Now, talking about the trunk, so also the dorsal side of the body. Do you see the dorsal side of the body, which has olive green, which is olive green with dark irregular spots, right? So this is the dorsal side being shown, and on the lower side, the ventral side will be pale yellowish in color. Well, in exam, we are just going to get a black and white picture, so it doesn't matter. All right. So these are the hind limbs and these are the forelimbs. Hind limbs are webbed. They are longer. They are stronger and helps in leaping. Is that clear? They help in leaping. See, the forelimbs and hind limbs, they help in swimming, walking, leaping and burrowing. Hind limbs end in, so they have 5 digits. While the forelimbs, guys, these will have 4 digits. Is that clear? So hind limbs are larger and muscular than the forelimbs that end in 4 digits. Feet are webbed. So webbed feet. Is that clear? Alright, moving on to anatomy. So this diagram shows the digestive structure. See, it starts with mouth, of course. This is esophagus. Esophagus will lead into the stomach. And stomach will lead into intestine. Intestine is going to be small here because it is a carnivore. And we know carnivores have a smaller intestine. And see, this is rectum, and then rectum and urinary bladder and reproductive structure. They will all open in a common chamber known as cloaca. This was a question already. There was a question here. All right, let's see here. Yes, please. So this is the urinary bladder. So cloaca is a common opening, so common chamber where 3 openings are going to be present. One is of urinary bladder, second is of the rectum, and third is of the reproductive structure, and third is of genital pore or reproductive structure. Are we clear? So 3 things are together going to open in cloaca, and then cloaca commonly opens outside through cloacal aperture. Are these structures clear? Then look at the glands. Look at the glands. So this is this brown colored structure. This is liver, again 2 lobes. Then below the liver, we have gallbladder, which is going to store bile. Liver produces bile for the emulsification of fats. Gallbladder is going to store bile and release it as and when necessary. Is that clear? Then this is the heart, and heart is of course going to be three-chambered. These are the kidneys. Kidneys have fat bodies over it, and the tubes starting from the kidneys are called ureters. So are you clear with the labelings of this diagram? The labelings are clear to all. Now let's see the diagram of male reproductive system. So in males, be it humans or be it frog, the reproductive system converges with the excretory system, right? So you will see this is testes, the reproductive system. The main part is testes, right? And above the testes, we have these fat bodies. So there is a pair of testes. All right. So from testes will arise some seminiferous tubules also, and they are going to release. See here. Here. See. So male will have a pair of ovoid testes, then a double fold of peritoneum called mesorchium, which connects it to the kidney. Then vasa efferentia 10 to 12, they will arise from the testes and they will open on the side and open into the Bidder's canal. So see here. So these are the vasa efferentia, written how many? 10 to 12 in number, and these vasa efferentia will open into the Bidder's canal. This is the Bidder's canal. So Bidder's canal will be connected to this urinary canal, which will shift the sperms into the excretory tube, right? And from here will arise the urinogenital duct, also known as the ureter. So sperms will be carried outside through this. Are we clear? Are we clear? And they open in this common chamber known as a cloaca. Well, in males, there is no separate reproductive opening, right? So there is just rectum and urinogenital duct which will open in the cloaca. While in case of females, guys, in case of females, urinary and excretory structures are totally separate, right? So see, these are the ovaries, these irregular structures, these are the ovaries, and they are packed with many ova. So ova will be released in the coelomic area, and this is from where the oviduct, that is the fallopian tube, is going to pick the ova, right? Through fallopian tube. So first is ovary, then in the coelom, it goes into the fallopian tube. From fallopian tube, guys, from fallopian tube, it will be passed out through cloaca, through this into the cloaca. Are we clear? So here cloaca will have three openings. One, that of ureter. See, one of ureter, another of fallopian tubes, this reproductive duct, and third of urinary bladder. So common opening will be cloaca. Common chamber will be cloaca. Are we clear? Yes, guys. Are we clear here? So no functional connection with kidneys in case of female frog, right? And now fertilization is going to be external. So guys, with this we complete the structural organization or the structure of frog. Now moving on to the third unit, that is of cell. Let's see the diagrams here. What are the diagrams given in this chapter? So this figure 8.1 shows different shapes of the cell. See, look at the shape of RBC. Then WBC, we have different types of nuclei in different shaped nucleus in WBCs depending upon the type. Then this is columnar epithelium cells, long and narrow. This is highly branched and long cell. The longest cell is a nerve cell or a neuron, right? Then this is a tracheid, elongated, and this is the mesophyll cell, round and oval with so many chloroplasts. Alikya is feeling stressed that I'm forgetting everything. Alikya, this is how we all feel, that we are going to forget. But the more you think like this, the more there are chances that you will actually forget. So stop panicking. I know easy said than done, but still, you will have to calm down yourself. Okay, take deep breath and relax, because see, we have good memory. We all have good memory, right? And our brain always supports us. Our memory never Ma'am, what about cockroach? Sankalp will come on to the cockroach. Wait. So our memory will come back at the right time. So when you see that question, just take a deep breath, relax, and then think about it. You will be able to recall everything. Okay, don't worry. But the more you think about, so you have these negative emotions, you may forget. Is the paper easy? I did not set the paper, but I just know that we are all prepared for whatever type of paper they set in. But I don't think they are going to change the level of the paper. The level of the paper is going to stay the same. Level of the paper is going to stay the same. Okay, all right. Moving on to the structure of prokaryotic cells. See here, some prokaryotic and eukaryotic cells are shown in this figure. So this is a typical eukaryotic cell. The size is 10 to 20 micron meters. See, you see membrane bound structures here, Golgi body, ER, right, nucleus, then mitochondria. So we see multiple structure. These are vacuoles and so on. So this is a eukaryotic structure. So a typical bacterial cell with lots of flagella around it, 1 to 2 micron meter. These small dotted structures, PPLO, 0.1 micron meter, and then viruses even smaller than bacteria. That is why we say virus passes through bacteria proof filters. So look at the size. It is 0.02 to 0.2 micron meter. Now moving on to the next diagram given in this chapter, which is of eukaryotic cell, the detailed structure of plant cell and animal cell. Now how do we identify? So there is a large vacuole. Such large vacuole is present only in the plant cell. So this is a plant cell. So this is a vacuole. This is a plasma membrane. Then outside the plasma membrane, there's a cell wall. If you go inside, this is a cytoplasm, right? This is all cytoplasm, right? Then there are peroxisomes. Microtubules are shown in the form of a line. Look at this, plasmodesmata. These connections are plasmodesmata. They are only found between the plants. Then ER, without ribosomes. This is SER. This is smooth endoplasmic reticulum. While the vesicles present near ER, they should be lysosomes, and then ER with ribosomes are RER. This is the nucleus, the body, small spherical body within nucleus is nucleolus. Then another structure which is present near nucleus is Golgi apparatus. Look at the nuclear membrane, and that's it. And this is the mitochondria, and these small dots, they represent the ribosomes. What type of ribosomes should be present here in the cytoplasm? ATS type. Is that clear? While mitochondria and chloroplast, this is the chloroplast, will have 70S type of ribosomes. Now look at the diagram of animal cell. We don't see a very large vacuole here. It is an animal cell. Is that clear? So see, this is the nucleus, the nuclear envelope, the rounded body is called the nucleolus. Then this is how they have shown the SER. See, RER is always associated with the nuclear membrane. This is the cytoplasm. Look at these ribosomes. Which type of ribosomes? Again, ATS. And in mitochondria, we will have 70S. We don't have chloroplast. So look at the diagram of mitochondria. This is also mitochondria. These two perpendicularly lying structures are centrioles. Together they will form the centrosome. Are we clear? And these outgrowths of the plasma membrane to increase the surface area, this is known as microvilli. Are we clear? Now moving on to the next diagram, which represents a fluid mosaic model of plasma membrane. See, our plasma membrane is majorly made up of bilipid layer and proteins. So this is the lipid molecule. This is the tail. Inner side is the tail, outer side we have the head, right? And in between we have these proteins. These are all proteins, integrated partially or completely, right? So these are all proteins. Hence this is which type of lipid dominates the plasma membrane? It is phospholipid. So see, phospholipid bilayer, integrated proteins. So internal proteins or external proteins. While there is sugar associated with both lipid and protein, so these are the sugar molecules, right? And in between the plasma membrane, the second type of lipid which is commonly present is cholesterol for stabilizing the structure. So one is phospholipid, second is cholesterol, which are the most important fats for the plasma membrane. Is that clear? All right. Moving on to this structure, this represents the endoplasmic reticulum. Do we see this is nucleus and this is the endoplasmic reticulum with ribosomes attached. These are ribosomes, hence this is RER, and the one without ribosomes is SER. Clear to all? And look at these cisternae. This is that of Golgi apparatus. These are the vesicles getting pinched off, vesicles. So one is the cis side, other is the trans side. Cis is the forming side and trans is the maturing side. Is that clear? Now, look at the diagram of mitochondria. The outer membrane, inner membrane. We know mitochondria is double membraned structure. It is double membraned structure. There are only three structures which are double membraned. Nuclear membrane is double walled, mitochondria, and second is chloroplast. So outer and inner. Now, do we see the inner wall is highly folded on inside, and these folds are going to form cristae. These are the folds of the inner membrane. Folds of the inner membrane. Yes or no? What do these do? These folds of the inner membrane are going to increase the surface area, right? And cristae is a place where electron transport system is going to run. So maximum energy production will occur here due to oxidative phosphorylation. Now the space between the outer and inner is known as intermembrane space, also known as perimitochondrial space. Yes or no? And the link reaction. Yes or no? Is that clear? So read this line. The inner compartment filled with dense homogeneous substance known as matrix. So matrix is a dense homogeneous substance. Clear to all? These are the powerhouses of our body, of the cell, sorry. Now, talking about the chloroplast, again, double membrane structure, outer and inner. There are these rectangular structures called thylakoids, and they stack up to form grana, right? This is the location for light reaction. This is where light reaction occurs. And connecting the two grana are called stromal lamellae. Again, light reaction. Is that clear? Now, the space around it is filled with stroma. This outer space is known as stroma. Stroma is the location or the site for dark reaction. Biochemical C3 cycle occurs here. Is that clear? Now, the type of light reaction which occurs in thylakoid or granule membrane, what do you call it? Non-cyclic electron flow leading to non-cyclic photophosphorylation. While in stromal lamella, it is of cyclic type. Yes or no? Yes or no? Is that clear? I'm going to complete this class. I'm going to complete the diagrams, guys, in this session. So as long as the diagrams go. Now, talking about ribosomes. So ribosomes also known as pallid particles because they were discovered by George Pallid, right? And each ribosome, whether it is 70S or 80S, is made up of small subunit and large subunit. So if we talk about the 80S ribosomes, which are present only in eukaryotes, so 80S will have 40S ribosome as a small subunit and large subunit will be 60S. While in case of 70S ribosomes, the prokaryotic type, large subunit will have 50S value and the small subunit will have 30S value. Is that clear? Ribosomes are further made up of two components, rRNA and proteins. Please tell me these points are clear, right? Now, see the cytoskeleton. This is the structure of cilia or flagella. So cilia or flagella will have 9+2 arrangement. So there are 9 tubules on the outer side. See, 9 microtubules, and they are of doublet nature. 1, 2, 1, 2, 1, 2. So 2 microtubules clubbed together to form the outer this microtubule. Is that clear? While there are 2 in the center, so central microtubules are also 2. So 9 on the outer side, 2 in the center, hence 9+2 condition. Do you see all the peripheral microtubules? They are connected through interdoublet bridge. Interdoublet bridge, also known as linkers, right? They are also connected to the central sheath. Central sheath covers the central microtubules, and we see that the outer ones are outer peripheral tubules. They are attached to the central sheath. This is through radial spoke. How many radial spokes? 9. Is that clear? And this is the plasma membrane. The outer layer, this is the plasma membrane. Rest everything within that is called exoneme. So exoneme forms the central part, which has 9+2 arrangement of microtubules. Is that clear? This is cilia and flagella. Now see that structure of nucleus. So this is the nucleoplasm, the matrix, the small spherical body called nucleolus. It can be 1 to 2 or max 3. Then nuclear pores, where the outer and inner nuclear membrane fuse. And this is the nuclear membrane. Again, these are also outer and inner 2. Is that clear to everyone? And inside lies the chromatin material or the genetic material. See the structure of a chromosome. So this whole, this one area, this is one chromatid, right? This is one chromatid. While in this diagram, there are 2 chromatids shown here. There are 2 chromatids shown here. This is the second one. So both of them together form the sister chromatids. They are connected to the same centromere, and around the centromere, guys, is a present small disc-like structures. Disc-like plates or structures called kinetochores. How many kinetochores around the centriole? 2, 1 on each side. And this is the site for attachment of spindle fibers. This is where spindle fibers attach. Site for attachment of spindle fibers. Please tell me these points are clear. Right? Now look at the different type of chromosomes. Now based on the position of centromere, see what is the criteria? Types of chromosome. The criteria is position of centromere. When the centromere is right in the center, so both P and Q arms are equal, this one is known as metacentric. This one is called metacentric chromosome. Then when the centromere is slightly away, right? When it is slightly away, it is known as submetacentric. Slightly, for S for slightly away and S for submetacentric. While in third case, centromere is quite away from the center. This is called away from the center. We call this as acrocentric. So one arm is very long, other arm is very short. And then towards the tip, we have this telocentric. Is that clear to all? So 4 types. This question has already been asked, guys, as a PYQ in match the following format. So be ready for the diagram based question also. Now, with this we complete cell. Moving on to the next chapter, that is of biomolecules. We don't have any diagram in here, but we'll go through some tables. So this table representing the percentage of elements present in Earth's crust and human body. So you should know the value of at least top 4. Top 4. So one is oxygen, second is carbon, then hydrogen, then calcium, and here we have sodium, right? OC-Kana. Hydrogen here is less. So you should know at least the top 4. Is that clear? And then in case of human body, it is again oxygen, it is carbon, then we have hydrogen, and we have nitrogen. Is that clear? So top 4 should be remembered by you. Then next table, which is important here. Okay, before we go on to the table, this is the formula of the Zwitter ion. Very important. Which molecule is known for forming the Zwitter ion? That is amino acid only. What is Zwitter ion? So such a radical which has both positive and negative charge occurring simultaneously. At a particular equilibrium, it will have both positive and minus charge occurring simultaneously. This is called Zwitter. Zwitter means hybrid, right? So only amino acids can do that. So see, both amino and carboxyl groups are ionizable in this case. Is that clear? Now moving on to this page. Yes, look at the structure of glucose, C6H12O6. Look at the location of each group, where is OH present, and then ribose. Ribose is an RNA sugar. See the formula as well as the diagram. Then 3 amino acids have been given in here. See, hydrogen, carboxyl and amino groups are constant. The R group is variable. In case of glycine, R group is H. While in alanine, R is equal to methyl group. While in serine, R is equal to CH2OH. Hence, serine is alcoholic. While both glycine and alanine, these are neutral amino acid. Yes. Now see here. The basic structure of a fatty acid. So fatty acid means it will have carboxyl group. This is an example of palmitic acid. Palmitic acid will have C16 including this. So C, CH3, CH2, 14 and then O. So total 15 in this chain, side chain, and 16th of the carboxyl group. So including carboxyl group, how many carbon atoms are present in palmitic acid? It is C16. Read the question properly, whether it is asking for including carboxyl group or excluding. Are we clear? Now, see the very simple alcohol, that is trihydroxypropane, which is also known as glycerol. Now, all these 3 hydrogens are replaceable. They can be substituted with the fatty acids. See the structure of a triglyceride. So this is glycerol. Told you? These hydrogens are replaceable. So they have been replaced by different fatty acids. This is a triglyceride. So there are, in triglyceride or monoglyceride or diglyceride, there is always one glycerol. So whether it is mono, di or tri depends upon the number of fatty acids. There are 3 fatty acids and the fatty acids are different. Is that clear? Now see, this is a modified, this is a conjugate lipid. So there is a phosphate group attached, phosphoric acid in fact attached. Hence it is a phospholipid. Example is lecithin, and which is commonly present in the plasma membranes. And see the structure there, see the group here. So this is a phospholipid. Then this is cholesterol, second most abundant fat present in the plasma membrane, which provides stability. Now these are the structures of nitrogenous bases. Look at this. This is adenine, which is a purine, and this is uracil, which is a pyrimidine. So purine is a 9-membered structure, double ring, while pyrimidine is a 6-membered structure, single ring. And adenosine. So when we add sugar to adenine, it becomes adenosine. So this is the sugar and this is the adenine, right? Similarly, when we add sugar here, it will become uridine, right? And when we add phosphoric acid to it, it will become adenyllic acid, and similarly uridylic acid. So nitrogenous base with sugar, it will become nucleoside, and with phosphate or phosphoric acid, it will become a nucleotide, and nucleotide is the monomeric unit of any nucleic acid. Are we clear? Very important table. Secondary metabolites. See, pigments. Which one are pigments? You can easily get this question in the form of match the following. Carotenoids and anthocyanins, they are pigments. Alkaloids, example is morphine and codeine. Terpenoids, easy to remember, monoterpene, diterpene. Essential oil, easy to remember, lemon grass oil, rose oil. Toxins, again, put some effort, heparin and ricin. Lectins, concanvaline A. Drugs, vinblastine and curcumin. Curcumin comes from haldi and vinblastine comes from vinca rosea, important for blood cancer treatment. Polymeric substances, rubber, gums and cellulose. Is that clear? Yes, please. Now, another important table, average composition of cells. How do we remember? We remember it as pinacally. Pinacally. The most abundant is water, of course. Other than water, we have pinacally. P for proteins, 1 is water, second position 2 proteins, Na for nucleic acid, C for carbohydrates, see the percentage, L for lipids, and I for basically ions. So you should be able to identify. See, if you remember this order, this is the decreasing order in which the components are present. If you remember this decreasing order, you can easily match, but also keep some idea about the values in case they ask you in the statement format, right? So water 70 to 90, proteins 10 to 15, then nucleic acid 5 to 7, then 3 to 1. Can you remember this? Now, another very important table, we usually get a question out of this table, one or the other question. So function of proteins. So proteins and functions. Collagen, already been asked. So intercellular ground substance. Trypsin is an enzyme, protein digesting enzyme. Insulin is a hormone, right, which lowers the blood sugar level. Antibodies, we know, they fight infections. Receptors for sensory reception of all different stimuli. And GLUT4, already been asked, enables the glucose transport. GLUT4 is glucose transport. Are we clear here? Very important table. Now, coming here, this is the structure of glycogen. So glycogen is a highly branched structure. What is the monomeric unit, guys? Monomeric unit of glycogen is glucose, right? So on the right hand side, see, this is the left hand side, this is the right hand side. Remember, right hand side is always reducing and left hand side is always non-reducing. Is that clear? Yes, please. Now, glycogen will also form a complex with iodine, by the way. Is that clear? This is the structure of glycogen. Now, moving on to the structure of proteins. We have primary, secondary, tertiary, and quaternary. Look at the primary structure. It is one direction, 1D, right? Primary. So, basically, it is a linear structure. They are arranged in one line, which shows you the number of amino acids and the sequence of amino acids. Oh, wow. Thank you. You know, green hearts give me as much energy as green color gave it to Hulk. No matter how sad. All right, guys. Moving on to the secondary structure. Secondary structure is two-dimensional structure, right? So we have two types. One is alpha helix, second is beta pleated. Is that clear? So alpha helix structure and beta pleated. Remember, alpha helix is always right-handed helix. It is always, proteins are only right-handed helices. Clear to all? Is that clear to all? All right. Next is tertiary structure or three-dimensional structure, which is very important for biological activity. Biological activity, like in case of enzymes. So enzymes have three-dimensional structure and quaternary structure, like in case of hemoglobin. So quaternary structure is present in such proteins which have more than one polypeptide chain, right? More than one polypeptide chain. So these are the four different structures you should be able to identify. And now we have this graph. So you look at this graph, concept of activation energy. So progress of reaction taken along the x-axis and potential energy has been taken along the y-axis. This is the transition state. This is the energy possessed by the substrates. This is the energy possessed by the products. So this is transition state, very transient and temporary and unstable, right? This is the activation energy without enzyme. That is why it is more, while this is the activation energy in the presence of enzyme. Hence it is less. You should be able to label these graphs carefully. Now, okay, we have another graph showing the effect of pH and temperature. What type of graphs do we get? We get bell-shaped graphs. Now remember, on the y-axis, we are taking the enzyme activity. On the x-axis, we are either taking temperature or pH. And this, where the maximum activity is observed, this pH is called the optimum pH or optimum temperature. Now, again, when we study the effect of concentration of substrate, this is y-axis. Along the y-axis, we take the velocity of reaction, while here on x-axis, we are taking the S in square brackets. This represents substrate concentration, right? So what type of graph do we see? Rectangular, hyperbola. Note down the values Km, Vmax by 2. Is that clear? Now, so with this, we complete this topic also. moving on to the last chapter of unit 3, guys, that is cell cycle. The first figure shows the various parts or phases of life cycle, of cell cycle. So G1, S and G2, this represents the interphase. This all represents the interphase, right? This is interphase. The resting phase, basically the preparatory phase. And the actual dividing phase is the M phase, which is further divided into PMAT, prophase, metaphase, anaphase, telophase. This is all karyokinesis, and the last phase is cytokinesis. Karyokinesis is nuclear division, cytokinesis is the cytoplasmic division. Clear? All right. Come on to the diagrams here. All right. So these are the phases of mitosis, the equational division, the early prophase, where the chromatin fibers have started condensing into chromosomes. Nuclear membrane has started disintegrating, but not completely done, right? Here in the late prophase, we see the nuclear membrane has shriveled up. The chromatin material has also condensed to form the chromosomes. Is that clear? So this is the prophase. Then in metaphase, so this is metaphase, this is transition into the metaphase. So basically still prophase going on. These are the centrioles moving towards the poles. These are the astral rays. These are the asters. These are the centrioles, right? This is the spindle fibers. And now when all the chromosomes come to lie in the center, this center is called the equatorial plate or the metaphasic plate. Now when they all lie on the equatorial plate, this is indication of metaphase. Are we clear? Then in case of anaphase, sister chromatids are going to split. So centromeres split and sister chromatids separate. Sister chromatids separate, chromatids move towards the opposite poles. Do we see them moving towards the opposite poles? This is anaphase, right? And when the chromosomes which have moved towards the poles, the nuclear membrane regenerates, then ER, Golgi body, mitochondria, all that disappeared in the plasma, in the prophase, it has all come back. See, nuclear membrane redevelops. All these structures reform. This represents telophase. So in telophase, you see two daughter nuclei within the same cell, two daughter nuclei. So now there has to be cytokinesis. If there will be no cytokinesis, it will result in polyploidy condition. We have read that line in genetics, yes or no? Right. So now the furrow formation, because this represents an animal cell, so furrow formation has started occurring for the cytokinesis to take place, right? So see, appearance of furrow in the plasma membrane in case of animal cell. That's how cytokinesis occur. Please tell me these points are clear. And now see the diagrams of meiosis. See, in prophase 1, so basically two homologous chromosomes lie side by side. So that happens only during meiosis, synapses. We call it synapses, yes. So in prophase 1, you will be able to see the homologous chromosomes. Homologous chromosomes showing synapses. Particularly, synapses occurs within which phase? Zygotiny phase, right? All right. Now, in metaphase, unlike mitosis, in metaphase 1 of prophase 1, metaphase 1 of meiosis 1, there are two equatorial plates, right? And each of the homologous chromosome lies on two different plates. See, these are two homologous, one on the upper, other on the lower equatorial plate. Similarly, in case of red ones. Then in anaphase 1, homologous chromosomes is separate. Splitting of sister chromatids does not occur, right? And this is telophase, when and after the telophase the daughter cells have been formed. Is that clear? While the meiosis 2 represents the mitotic part only. So meiosis 2 is just like mitosis. So we have one out of each homologous pair, right? One blue here, one blue here, one red here, one other red here. So again, they've started moving towards the equatorial plate. So this is the metaphase 2. Then sister chromatids separate here. sister chromatids are going to separate in anaphase 2 phase. And finally, there are 4 daughter cells formed. This is telophase. Please tell me these points are clear. Yes, guys, are we clear till here? Everyone? All right, let's move on to unit number 4. We are going to revise the first chapter here of plant physiology, unit 4. Yes, please. Starting with photosynthesis. So guys, this diagram represents a Priestley's experiment where he identified that oxygen is important. Sorry, oxygen is evolved. So he did this bell jar experiment. See here. He did this bell jar experiment. And see, a bell jar experiment. So when a mouse and burning candle were kept together in this bell jar, after a while mouse died and the candle extinguished, right? But when he kept this mint plant, which plant did he use? Mint plant was kept along with the mice and the burning candle. Both of them stayed alive. Mouse was happy, the candle was burning. So what did he conclude? What did he conclude? That plants restore the air, whatever breathing animals and burning candles remove. Is that clear? Right. Now, moving on to the next diagram here, or that is of chloroplast. Do we see an outer membrane, inner membrane? So double envelope structure. Then we have these thylakoids. Each part is called thylakoid. The stack is known as grana, right? Now connecting the two grana, we have fret membranes, also known as stromal lamella. This outer matrix is called stroma, where dark reaction will take place. Now we know, be it mitochondria, chloroplast, they have their own ribosome, 70S, prokaryotic type. They also have their own DNA, again, prokaryotic nature, circular. So what does this indicate? This indicates their semi-autonomous nature. So they can make some of their own proteins by themselves, semi-autonomous in nature. Now because it is going to produce food which will be stored in the form of starch, there are some starch granules, lipid granules as well. So this is the structure of chloroplast. Are we clear? Very important graphs, very important. See the first graph here. So this is the absorption spectrum. First of all, this is the absorption spectrum. So which wavelength of light is absorbed by different pigments, that's depicted here. The absorption or absorbance of light by chloroplast pigments. So the first peak, this blue one, this is of A. The second peak, this is of B, and A is brighter blue green. Then chlorophyll B is yellow green, this is B, and these so many spikes indicate the carotenoid. Is that clear? All right. Now we also observe that the same place where chlorophyll A showed spike in absorbance, we see a spike and the rate of photosynthesis also. Like see, there are two spikes seen here. So that means whenever chlorophyll is absorbing more light, it is showing more action. This is the action spectrum. What is the action performed here? That is performing photosynthesis. So whenever more light is absorbed, more action, more photosynthesis is also done. Is that clear? Now look at this diagram. This diagram represents the light harvesting complex. The surrounding molecules are the pigment molecules. In the center we have reaction center. Reaction center is nothing but chlorophyll A, and then photons of light are being absorbed, and then electron will be ejected from the chlorophyll A, and there will be a primary acceptor. Now that depends on the photosystem. Which photosystem is it? See, this represents a Z scheme of light reaction. Z scheme is the non-cyclic form of electron flow. Non-cyclic. So in Z scheme, guys, first of all we have PS2 and then comes PS1. In PS1 the maxima is at P700 and Maxima is at 700 nanometers. So we call this reaction center P700. In PS2, we have maxima coming at 680 nanometer. So we call it P680. So whenever light falls, electron gets ejected. See, it is taken up by the electron acceptor. Electron acceptor of PS2 is PP, pheophytin, while in case of photosystem 1, it is ferredoxin, right? Then from PP, it gets to the PS1 through electron transport system. And we know what comes in the electron transport system: plastoquinone, cytochrome B6F, then plastocyanin. These three will be the components here. And because the electrons once lost are not coming back, that is why we also call it non-cyclic. And look at the structure. So it goes uphill, then downhill, and then uphill again. So it forms Z shape, and hence we call it Z scheme of electron flow. Now we need to compensate the loss of electron. So PS2 is compensated by water splitting complex. So water splitting complex is associated with PS2. We don't need any such complex for PS1, because PS1 is compensated by the electron coming from PS2. So who compensates for PS1? PS2. Who compensates for PS2? It is the photolysis of water, splitting of water. Is that clear? All right, guys. Now moving on to the cyclic photophosphorylation. Why here in this only PS1 is involved? Secondly, the electron acceptor is just ferredoxin, and there is no NADP reductase, right? So NADPH is not formed. So the electron once lost will come back to the photosystem 1. So hence it is cyclic phosphorylation. Where does this run? This runs in the stromal lamellae. This occurs in the stromal lamellae, while the previous one, that is non-cyclic or Z scheme, it occurs in granule lamellae. Or thylakoid membranes. Are we clear? Yes, guys, are we clear till here? Now, this is the ATP synthesis through chemiosmosis. This is the thylakoid membrane being shown. Outside we have stroma, and do we see this is PS2, this is PS1. I've shown the whole electron flow already. So this is representing the non-cyclic or the Z scheme of electron flow. This is the Z scheme of electron flow, right? Now, because of water splitting, there are so many H positive ions. Also plastoquinone, which is a hydrogen carrier, proton carrier, it is pumping protons from stroma into the lumen. So lumen will have high electrochemical potential, while the stroma will have low. So H positive ions will move out through this. This is called ATP synthase. Now ATP synthase has two components, CF0 and CF1. CF1 is towards the stromal side. CF0 is embedded within the thylakoid membrane. Is that clear? And see, for every 4 protons pumped out, 1 ATP will be generated. This is chemiosmosis hypothesis. This is chemiosmotic hypothesis. Are we clear? Now, moving on to the cycle, that is C3 cycle. Look here. So first of all, C3 cycle or the Kelvin cycle. What are the 3 steps? One is carboxylation, second is the reduction, and third is regeneration. You must know them in a particular order. So the order is CRR. Is that clear? CRR. Carboxylation, reduction, and regeneration. Alphabetical order, red and reg. Clear? Now, during this, see, there are first, which is the carbon dioxide acceptor. This is a primary CO2 acceptor, that is RUBP. Ribulose 1,5-bisphosphate. How many carbons? 5 carbon compound, 1 carbon from CO2. What should it form? It should give us 6 carbons, but instead of forming 1 6-carbon compound, it is forming 2 molecules of 3-PGA, 3-phosphoglycerate, right? Which contains 3 carbon. 3 into 2, 6. So that's how it is balanced. Now see here, why do we call Calvin cycle as C3 cycle? Because the first stable compound here is a 3-carbon compound. It is a 3-carbon compound. Are we clear? Right. Then 3 PGA will get reduced in the presence of 1 ATP and 1 NADPH and form triose phosphate. So 2 will form, of course, 2. Is that clear? And then again, the 2 molecules of triose phosphate will combine together to form or regenerate RUBP at the expense of 1 ATP. Now from triose phosphate, again it has 3-carbon compound. How many are formed? 2. We are going to take 1 carbon out of it. So ultimately 6 such cycles should run. So 6 such cycles should run to form 1 molecule of glucose. 1 molecule of glucose. Is that clear? Now see the expenditure here. So for 6 carbon molecules to be fixed, 1 glucose molecule will be produced, and at the expense of 18 ATP and 12 NADPH. What goes in, what comes out, you should be very, very clear. There are so many questions coming around this topic. Is that clear? So this represents the Hatch and Slash pathway, that is the C4 cycle, cycle. So unlike in C3 plants where Rubisco, see Rubisco, where was Rubisco used, by the way? Let's go back. Rubisco is catalyzing the first reaction, that is carboxylation. This is where Rubisco is used. It is involved in carboxylation. Where is the cycle occurring? It is occurring in the mesophyll cell. It is occurring in the mesophyll cell, right? But in case of C4 plants, look here. In case of C4 plants, where C4 cycle will occur first and then C3 cycle. See, C3 cycle has to occur because food production will occur only through C3 cycle, but it will be preceded by C4 cycle which occurs in mesophyll cell. So mesophyll cell will have peptase. Mesophyll cell will have C4 cycle. C3 cycle and Rubisco. Rubisco is present in bundle sheath cell. This is where C3 cycle will occur. So you should know the pathway. Is that clear? All right. Now this table is important. Graph, the graph of light intensity affecting the rate of photosynthesis. Y-axis we take the rate and x-axis we take the light intensity. See, initially there is a linear relationship between the light incident and CO2 fixation at the low intensity. So A represents at low intensity of light. At low intensity of light, there is a linear relationship. That means you increase the light, the rate of photosynthesis also increases, right? But at higher intensity of light, at higher intensity, it does not show further increase, right? What is interesting, that light saturation will occur at 10%. So this is when the light has been saturated. This is light saturation. Is that clear to all? And after a while, if you still keep on increasing the light intensity, then the graph will also come downward because the chlorophyll will be destroyed because of intense light intensity, because of very high light intensity. Clear? So guys, with this, we complete the first plant physiology chapter. Moving on to the chapter number 2 of plant physiology, that is respiration in plants. Yes, guys. See, these are steps of glycolysis. It is a 10-step process which was discovered by, the pathway was discovered by AIMT, right? So 10-step process, anaerobic, occurring in cytoplasm, common to all organisms. So glucose changing into glucose 6-phosphate, then fructose 6-phosphate. You have to note down where ATP is being used. So till here, guys, ATP is getting used. So this is ATP utilizing phase. One, when glucose is changing into glucose 6-phosphate, and then when fructose 6-phosphate is changing into fructose 1,6-bisphosphate. So 2 ATP molecules have been used, 2 ATP used. Is that clear? Then this is called splitting when 6, so this is 6 carbon, 6 carbon, 6 carbon, 6 carbon. it splits to form 3 carbon each compound. Is that clear? And then after a while, it will start producing ATP. Which are the steps where ATP is produced? 1,3-bisphosphoglyceric acid changes to 3-PGA, and then 2-PEP changes into PEP changes into pyruvic acid. So because the same is occurring on this side also, we are just multiplying it with 2. So 4 molecules of ATP are yielded. This is ATP yielding phase, and during which 4 ATP molecules are produced. So what is the net gain? That is 2 ATP. Net gain of direct ATP is 2 ATP. Is that clear to all? Right? All right. Moving on to this is the anaerobic pathway. So for till here, till pyruvic acid, we have glycolysis. This is glycolysis. Now, fate of pyruvic acid could be forming lactic acid in case of anaerobically respiring muscle cells, right? Or it can also form ethanol plus CO2. So this is called alcoholic fermentation, and see, this will be known as lactic acid fermentation. Depends upon the cell, either of the two will occur. Is that clear? All right. Now, moving on to Krebs cycle, which is also known as citric acid cycle. So first of all, pyruvate will change into acetyl coenzyme A, and this is known as link reaction. So link reaction will also occur in the mitochondrial matrix. So once the acetyl coenzyme A is formed, it will enter the it will enter the Krebs cycle, that is also called citric acid cycle. Why citric acid? Because the first compound formed during the Krebs cycle is citric acid. So OAA, oxaloacetic acid, will join the acetyl coenzyme A to form citric acid, and that's how the cycle will start. So citric into isocitric, isocitric to alpha keto. So whenever there is a step down in the carbon number, you will see evolution of CO2 occurring. So 5 carbon to 4 carbon, again carbon dioxide is being evolved. So this is one Krebs cycle. So from one glucose, we got 2 pyruvates, right? So one glucose will give you how many pyruvates? 2 pyruvates. So 2 of each. So you will run this whole thing twice, right? So whenever you're calculating something, just make it twice. Okay. So how many carbon dioxides have been released during the Krebs cycle? There are 2 decarboxylations. How many carbon dioxides? One here and other here. Then one carbon dioxide was also released during the link reaction. So in total 3 CO2. How many link reactions and how many Krebs cycles are running? 2 each, right? So 3 into 2, 6 CO2 are released during the complete oxidation of one glucose molecule. Is that clear? Then how many NADH? 3 NADH during the Krebs cycle, 1 NADH during the link reaction. Yes. Then also, how many FADH2? Just 1 FADH2. How many GTP? Only 1 GTP. But you have to twice it as per the deal. Is that clear? And last not the least, we have ETS here. First thing, you should know the name of the complexes. Complex 1 is NADH dehydrogenase, right? Complex 2, guys, is succinate dehydrogenase. Complex 3 is cytochrome BC1. Complex 4 is cytochrome C oxidase, and complex 5 is ATP synthase. And then you should know how the electrons move. First of all, you should be able to identify the complexes. Is that clear? All right. Then this represents the structure of ATP synthase, which has 2 components, F0 embedded in the inner mitochondrial membrane, and then F1 towards the matrix. Is that clear? Because on the outer side we'll have more that means in the perimitochondrial space, this is the perimitochondrial space, we'll have more H positive ions than in the matrix, low H positive ions, right? So H positive ions will move, this is the outer layer, this is the inner layer. Yes, guys. So this is how it is present, right? So these are where the complexes are present. So more H positive ions are in the perimitochondrial space. Are we clear? So they move into the matrix through this F naught particle, and F1 will show change in the in the it will help, F1 will help in the ATP formation. So for every 4 H positive ions passing in, it will form 1 ATP molecule. See, 4 H positive pass through F naught and it will move down the electrochemical proton gradient. Are we clear to everyone? Are these points clear? Right? And last but not the least, we have this important table showing the interrelationship among metabolic pathways. So what are the 3 different respiratory substrates? These are all the respiratory substrates. It could be fats, it could be carbohydrate, and it could be proteins. So we have seen the pathway of carbohydrates. So let's say if it is glucose, it is going to follow the glycolytic pathway and then link reaction and Krebs cycle, right? So after glycolysis and link reaction, after link reaction, it is going to form acetyl coenzyme A and enter the Krebs cycle. If we see the breakdown of fats, fats are made up of fatty acids and glycerol. Glycerol is going to break down to form DHAP and enter the glycolytic pathway and again form acetyl coenzyme A. Fatty acids, while they can be converted into acetyl coenzyme A, right? So all different respiratory substrates will enter the same pathway at one or the other point. Is that clear? Somehow. So point is, and similarly we see when proteins break down to form amino acids will form pyruvic acid and again form acetyl coenzyme A. So we see that acetyl coenzyme A is a common byproduct of no matter what respiratory substrate you're breaking down. Are we clear? Right? So then respiratory quotient, you should know the values, and that's it, guys. Then we have last chapter of plant physiology, plant growth and development. Look at this diagram, guys. This shows the germination and seedling development in the beans. So this is soil, this is hypocotyle, this represents the cotyledon, then seed coats are breaking down, epicotyle hook is formed, and that's how germination is occurring. Is that clear? Now, coming on to this represents the pollen tube growth in case of pollen grain germination. Yes, let's talk about the growth rates. So when with every division only there is increase of only one cell, this represents arithmetic growth rate, while in geometric the number gets doubled up with every division, right? Now, very important question which has already been asked, what type of growth is shown by embryo during the development? So embryonic development shows both geometric and arithmetic phases. So see, initially zygote is dividing with geometric progression, but eventually it will come down to arithmetic phase. So ultimately embryo shows both geometric and arithmetic phases. Is that clear? So this was a question already. Now see, the linear growth, that is the arithmetic growth. Arithmetic growth shows it is a constant linear growth when only one cell continues to divide and the other differentiates and matures. Is that clear? So we see a gradual increase in the number of cells. And what is the formula? LT is equal to L naught plus RT. While in case of geometric growth, guys, we see 3 phases. One is the lag phase, exponential phase, and then the stationary phase. And see the equation here. So it gives us S-shaped graph. So geometric will always give you S-shaped or the sigmoid graph, while the other one is going to give you a linear graph. Is that clear? For arithmetic, the shape of the graph is linear. For this sigmoid, for geometric we have sigmoid. So geometric will give us sigmoid, while arithmetic will give you the linear graph. Is that clear to all? All right. Now these two diagrams help us to compare the absolute and the relative growth rates. So see, absolute, the formula for absolute growth is final minus initial. It is final minus initial. So what is final minus initial in A? It is 10 minus 5, which is 5 centimeters square. While in leaf B also, it is 55 minus 50, that is 5 centimeters square. So the absolute growth for growth rate for both the leaves is same. But if we have to talk about the relative growth rate, there has to be a common point of comparison. So relative growth is equal to final minus initial upon initial, the common parameter for which we are comparing the two. For example, here it will be 10 minus 5 upon 5. So 5 upon 5, it is 1. Yes. Now in case of leaf B, it will be 5 upon 50. So what would the answer be? 1 upon 10. Do you see how the relative growth rate is different for both, right? It actually helps you compare. Now, talking about the next diagram, very important. So this has already been asked, guys. This is a question already. So meristematic cell. Meristematic cell is an actively dividing cell. So meristematic cell has to undergo plasmatic growth, differentiation, expansion and maturation to form the mature cell, right? Now they can leave these points 1, 2, 3, 4 as blanks and you have to identify. Now please learn it as such. On top we have PD and here we have EM. So plasmatic growth, differentiation, expansion and maturation. I know you will be easily able to remember all the four processes which convert a meristematic cell into the mature cell. But now the trick is you also have to remember them in a particular order. So plasmatic growth, then differentiation, PD and EM. Is that clear? Then expansion and maturation. Learn them in this particular order. So then a mature cell is formed and mature cell will undergo aging or senescence and then it will lead to death. Is that clear? Now in case meristematic cell does not undergo any differentiation, elongation and maturation, it will continue to divide right after the plasmatic growth. Is that clear? Right? But for maturation to occur, for differentiation, so these processes will help in differentiation, development completely. For it has to undergo differentiation, expansion and maturation. Now, look at these diagrams. These two diagrams show heterophily. That means heterophily is a type of plasticity. It is a case of plasticity. Now, this one is larkspur. Larkspur, while the diagram B represents the buttercup. See, we see two types of leaves. In different types of leaves in terrestrial as well as in water habitat. Do we see in water habitat they are highly dissected? That is buttercup, showing heterophily with environmental changes. According to the environmental change, we see heterophily in buttercup. While in larkspur, there are three examples given here: cotton, coriander and larkspur. Larkspur will show heterophily depending upon the phase of life, the stage of life. So look at the shape of the leaf in the juvenile stage and that in case of adult. So remember, at juvenile and adult, that case is for larkspur and this is the case for buttercup, which shows heterophily with respect to change in the environment. Is that clear? So how do we remember? So in case of water, the leaves are highly dissected because this is an adaptation to prevent water resistance. Is that clear? Otherwise, in any case, water is going to tear away the leaf. Now to avoid that, the leaves have already been modified as highly dissected. Is that clear? Buttercup. Now this diagram shows the phototropism. So the tip of the calyptyle is the source of oxygen and it moves towards the light. So where is actually oxygen produced? Oxygen is produced away from light. Okay. So this experiment was done. This was unilateral illumination of the calyptyle. This experiment was done by Darwin and his son Francis Darwin in calyptiles of canary grass, and that's how oxygens got discovered, not identified. Is that clear? Then this diagram is showing the apical apical dominance. Apical dominance means as far as the tip is present and tip continues to grow, the lateral development will not occur. Is that clear? So this is apical dominance. Then there are hardly any diagrams in this chapter. So next, guys, we have human physiology. Let's start with the last part of the session, which is human physiology. Yes, guys, all the serious students here, quickly give me a thumbs up. Are we all ready? Are all the chapters clear till now? All chapters clear? Yes, please. All chapters clear? I think it will take us another 2 hours and we'll be able to complete the syllabus of class 11, sorry. All right, guys. Let's start with, just a second. Yes, let's start with this. Chapter 14, that is breathing and exchange of gases. All right. Let's see the diagram here. So this is the diagram of human respiratory system. Human respiratory system, see, this is epiglottis made up of elastic cartilage, right? And it prevents the entry of food into the larynx. What is the function of epiglottis? We know it prevents the entry of food into, see, it's clearly written epiglottis. It is thin elastic cartilage called epiglottis, prevents the entry of food into the larynx and then into the trachea. Then let's see the larynx here. Larynx is the voice box, which is again made up of cartilage. Then larynx will lead into trachea. Larynx will lead into trachea, that is the windpipe. And see, it is lined by these C-shaped cartilaginous rings. We call them incomplete cartilaginous rings, right? Again made up of haline cartilage. Made up of haline cartilage. Now, all these so trachea will now divide into bronchus. So what is the location? It divides at the level of T3 vertebra. At T3, that is fifth T5. Sorry, it divides at T5. So fifth thoracic vertebra. So bronchus divides at, bronchi divide at T5 level, fifth thoracic vertebra level. Is that clear? So you have to remember this into right and left, and each enters into the each lung. So are we able to see the lungs here? Are we able to see the lungs here? This is the right and this is the left one. And lungs are packed with these small sac-like structures called alveoli, right? Now lungs are protected by two pleural membranes, double-walled pleural membranes, and in between the pleural membranes is present the pleural fluid, right? Now bronchus, when it enters into the lung, it further divides into like primary, secondary, tertiary bronchi, and then into the bronchioles. All these bronchi and bronchioles, they are lined by the C-shaped cartilaginous rings. Remember this line. See, repeated divisions up to secondary, terminal bronchioles and bronchi and bronchioles. They are all, and initial bronchioles, they are all supported by, see, from trachea to initial bronchioles, they are all supported by incomplete cartilaginous rings. Are we clear? Now, moving on to the next diagram. Again, already been asked, this represents the process of breathing. So when you see the ribs and sternum are already raised, right, what will it do? And the diaphragm contracted, this is going to increase the volume of thorax, right? When the volume increases, pressure will decrease and therefore air enters. So this represents inspiration or inhalation, right? So ribs contract, ribs raise and diaphragm contracts and becomes flattened. So which are the muscles which help in raising the ribs? It is external intercostal muscles. We are talking about normal respiration. External intercostal intercostal muscles. Muscles will contract and ribs and sternum will raise. Is that clear? Now, the lower diagram, this represents expiration or exhalation. So for exhalation, that is air to move out, the pressure inside the lungs should become higher. So pressure becomes higher here. Why the pressure becomes higher? Because the volume reduces. How? The ribs and sternum return to the original position. This is because of relaxation of intercostal muscles, external intercostal muscles. Diaphragm also relaxes, becomes arches, that means it becomes dome shaped. So because of all these movements, the volume is going to reduce. Volume reduces, so pressure increases. Is that clear? Is that clear? Now, table 14.1, you have to learn the partial pressures in mm mercury. So atmospheric air will have partial pressure of oxygen 159 and carbon dioxide is just 0.3. Alveoli, oxygen partial pressure is 104 and carbon dioxide is 40. In deoxygenated blood, oxygen is 40 and CO2, PCO2 is 45. In oxygenated blood, the partial pressure of oxygen is 95 and CO2 is 40. And in tissues, again, oxygen has 40 mm mercury pressure, while PCO2 is, partial pressure of CO2 is 45. Learn the values. Values will help you understand. So air will always move, or gas will always move from higher to lower pressure area. Now this shows the exchange, gaseous exchange, exchange of gases at the alveolar level and body tissues with blood and transport of oxygen and CO2. So see, this is the alveolus. We have just read what is the partial pressure of oxygen in alveolus, 104, and that of CO2 is 40. Now, pulmonary artery, which is bringing the deoxygenated blood, what is the partial pressure here in deoxygenated blood? Of oxygen, it is 40. Here oxygen is 104, in deoxygenated it is 40. So oxygen will move from higher pressure to lower pressure area. When oxygen has moved into the blood has become oxygenated. And what is the value of partial pressure of oxygen now? 95. Is that clear? Similarly, partial pressure of CO2 in deoxygenated blood is 45. In alveolus, it is 40. So CO2 is going to move into the alveolus. So we have removed CO2 and we have taken oxygen, and overall the blood has become oxygenated. You should see the values here. Now this blood, oxygenated blood, when goes to the tissues, we have just seen what is the partial pressure of oxygen in tissues. They are metabolically active. So oxygen has been utilized. Hence oxygen is just 40 mm. While partial pressure of CO2 has increased because as a result of respiration, CO2 has been produced. So see, higher CO2 in the tissue will move towards the blood and higher oxygen from the deoxygenated blood will move into the tissues. Again, gaseous exchange occurring here. Please tell me these points are clear. Then we have the diagram of alveolus here. So air in the alveolar cavity, we know alveoli is lined by squamous epithelial cells, one cell thick. Now this is an adaptation for diffusion. Squamous epithelium is the only epithelium across which diffusion can take place. Yes. And then we have blood capillary network around the alveoli. Again, blood capillary is also lined by the single layer of squamous epithelium. We call it endothelium. And it is a characteristic of epithelial tissue that there is always a basement membrane present. So, gaseous exchange will occur through these layers. It will cross the squamous epithelium of alveoli, then basement membrane, and then endothelium, right? So, these three layers, 1, 2, and 3, they together form what? The diffusion membrane. Is it clear? 1, 2, and 3, they form the diffusion membrane. What is the total thickness? It is less than 1 mm. And this is the diffusion membrane across which gaseous exchange will take place. Now this is the famous ODC, that is oxygen dissociation curve. See, on the x-axis we are taking the partial pressure of oxygen in mm mercury, and along the y-axis we are considering percentage saturation of hemoglobin with oxygen, right? Now, what type of graph do we get? We get a sigmoid curve. We get a sigmoid curve, always S-shaped curve. And whenever there is a sigmoid curve, there are always three phases. We have lag phase, we have log phase or exponential phase, and we have stationary phase. The maximum saturation occurs, see here. So this is in the lungs. So when does percentage saturation of hemoglobin? 100% saturation occurs within the lungs, right? So this 25% saturation, this is at the tissue level. Are you getting that? So now there are certain factors which can shift this curve towards the right hand side. So right hand shift will indicate the dissociation. It will favor the dissociation. It favors oxygen hemoglobin breakdown, dissociation. So where does dissociation occur? This is at the tissue level. So at tissues, guys, it takes a right hand curve, right? While at alveolar level, it will take a left hand shift. So see, in alveoli, what are the factors which alveoli? So basically LHS, left hand shift, it favors oxygen hemoglobin association, formation of oxyhemoglobin. So what are the factors which show which favor left hand shift and then which favor the right hand shift? Is that clear to all? Yes, guys. Is that clear to everyone? So this is ODC. Now moving on to the diagrams of body fluids and circulation. See here. Quickly, we have these pictures of formed elements. So this is your RBC. These are the cell fragments called platelets, and these 5 different types of WBCs. So in these WBCs, eosinophil, which has this bilobed nucleus. Do we see this bilobed nucleus here? Bilobed nucleus. We have S-shaped nucleus in basophil, and we have multi-lobed nucleus in case of neutrophil. So this eosinophil, basophil, neutrophil, these are all granulocytes. While monocyte and lymphocytes, these are agranulocytes. They don't have any granules. Look at the shape of nucleus in monocyte. It is kidney shaped and very large nucleus. While in B or T lymphocytes, there is a large nucleus occupying maximum area. Do you see? Nucleus is very large, occupies most of the space, right? So these are both agranulocytes. Now, moving on to this blood group and the donor compatibility. See, blood group depends upon the antigen present on RBC surface. So antigen and blood group is always the same antigen, same blood group, right? But antibodies which are present in the plasma, they are opposite to the antigen present. So antigen A, then antibody anti-B. The B antigen, anti-A antibody. If you have both the antigens, like in case of blood group AB, there will be no antibody here, and that is what makes AB as universal recipients. Now in case of blood group O, there are no antigens, and that is what makes them universal donors. Is that clear? But O individuals will have both the antibodies in their plasma. So look at the donor group for each blood group very carefully. Is that clear? Hemal, why are you spamming here? What is this? 1, 2, 3, 1, 2, 3. Yes, Hemal? Are we learning maths here? Still at that level of 1, 2, 3? Then you are at the wrong place. All right. Next, we have the structure of heart. Very important. See, the structure of heart. So we are talking about human heart, which is 4-chambered. So do we see right chamber, the right atrium? The upper chambers are called atrium. This is the right atrium. This is the left atrium. The lower chambers are called ventricles. So the larger ones, the right ventricle and the left ventricle. Are we clear? Then there is the septum present. So septum between the ventricles is called interventricular septum, right? And similarly, the area septum between the atria will be known as interatrial septum. Now this lower end is called apex, and while this whole area is called the base, right? Look at this. This is our sino-auricular node or the pacemaker. What is the location of pacemaker? It is present in the upper right corner of right atrium, right? Upper right corner of the right atrium is SAN. Then AV node, again within the right auricle only, both of them are present in the right atrium. SAN in the upper right corner, while AVN in the lower left corner, but both of them are in the right atrium. Are we clear? Then from the AV node, do we see that bundle of His is originating, going into this interventricular septum, getting split into the two branches and forming these Purkinje fibers. These branches are Purkinje fibers. Clear? Now these white structures, white thread-like structures present in the ventricles are chordae tendineae. These are made up of collagen. They are actually very hard. What are they holding on to? They are holding on to these valves. This valve is tricuspid valve and these valves are the bicuspid valves. These are the bicuspid valves. Bicuspid between the left auricle, left ventricle, tricuspid between right auricle and right ventricle. So this explains the function of chordae tendineae. They are going to hold the valves to prevent them from opening into the opposite direction. Is that clear? And also, do we notice there are these elevated muscular structures present? These are papillary muscles. These are the papillary muscles. So basically, chordae tendineae are attached to valves on one side and they are attached to papillary muscles on the other side. Is that clear? In left ventricle, we have 2 of them. And here we have 3. These are all papillary muscles. Is it clear? Now this is the aorta. This is the pulmonary artery. There are 4 pulmonary veins entering into the left atrium. This represents the largest vein, vena cava, the superior vena cava and the inferior vena cava. Are we clear? Alright guys. So with this, okay. Now the last figure of this chapter is that of ECG, electrocardiogram. It represents these different waves. P wave indicates the atrial depolarization. QRS complex, this shows the ventricular depolarization, and T about the ventricular repolarization. Are we clear? So it is the electrical activity, guys. P does not indicate the atrial systole. Please understand. Atrial systole will occur after atrial depolarization, right? Now, by counting the number of QRS complexes, you can also get to know the number of heartbeats per minute. So, QRS complex is equal to the heart rate. Number of QRS complex is equal to the heart rate. Is that clear? Now, here we see the double circulation. We have double circulation. So, the same blood entering the heart twice, one in the form of oxygenated, second in the form of deoxygenated. So see, this is your heart. So blood moving into the heart brought by the vena cava or the great veins. This is deoxygenated. This is deoxygenated. It goes into the right side and from right ventricle it goes into the lungs through pulmonary artery. So pulmonary artery is the only artery carrying deoxygenated blood. So from right ventricle to lungs and coming back to the left auricle. This is called pulmonary circulation. This part is called pulmonary circulation, which involves the lungs, right? Then similarly, blood from the left ventricle will go towards the body and from body, it will come back into the right auricle. This represents the systemic circulation. This represents the systemic circulation. Of course, the longer route, but it is around the body. Is that clear? So, oxygenated blood, deoxygenated blood. This is deoxygenated blood and oxygenated blood. I hope you understand. So, we have 2 pathways in double circulation. So, same blood is entering the twice. Are we clear here? Yes, please. Are we able to understand this much? So guys, with this, all the diagrams of body fluids and circulation are also done. Moving on to the next chapter, which is excretory products and their elimination. Excretory products and their elimination. See here, this is the human excretory system comprising of kidney, 2 pairs of kidneys. I mean, 1 pair of kidneys, 2 kidneys. This is the right kidney and this is the left kidney, right? There is renal artery bringing oxygenated blood to the kidney and this is deoxygenated blood being carried away through the renal vein. Now there is a gland present on top of kidney which is called adrenal gland on the superior side, right? Now see here. So each kidney is divided into two regions. The outer region is known as cortex, the inner region is known as medulla. Clear? Now from kidney arises the ureter. So there is one pair of kidney, one pair of ureters. So ureters are going to transport urine to the urinary bladder where it will be stored till it is evacuated, right? And then from urinary bladder arises urethra. Now in case of males, it is going to be the common duct for transport of sperms as well as urine. Hence it will be known as urinogenital, but not in case of females. Is it clear? And this is the urethral meatus. Please tell me these points are clear. Right? Now see the detailed structure, the longitudinal section of kidney. Already told you, outer structure is known as cortex, inner structure is known as medulla. Right? Now medulla is divided into medullary pyramids. These triangular structures are known as medullary pyramids. Clear? Now ahead of medullary pyramids, we have these renal papillae also. We have renal papillae. And from these pyramids or papillae arise these small ducts. These ducts are known as calyx. Calyx means ducts. So we have smaller ducts opening into the larger ducts. These are the larger ducts. And finally, the larger ducts will open into this broad area known as pelvis. This area is known as pelvis. See. This area, this broad area is known as pelvis, and from pelvis arises the ureter. Is that clear? So the area, that depressed area, that notch through which ureter comes out, that is called hilum. So this area is called hilum. Are we clear? Now the protective covering is present around the kidneys also, which is known as renal capsule. Also notice that some cortex, outer region is cortex, this is all cortex. So cortex is going in between the medullary pyramids. So all these structures, so this cortical area going into the medullary pyramids, this is called renal column or columns of Bertini. Or columns of Bertini. Are we clear? Please let me know that points are clear till here. Everybody? Yes, please. Yes, guys. Is it clear to everyone? Yes, please. Yes. All clear? All right. Let's proceed then. So this is the structural and functional unit of kidney called nephron or uriniferous tubules, right? So nephron has how many parts? So first part is glomerulus. So parts of nephron. So what are the parts of nephron? Parts. So there are around 1 million nephrons in each kidney. So first part is glomerulus and the second part is the renal tubule. The second part is the renal tubule. And in renal tubule, we have Bowman's capsule, we have PCT, we have loop of Henle, and we have DCT. Is that clear? So first of all, renal tubule, renal artery particularly, renal artery which brings oxygenated blood gets broken down into afferent arteriole. So afferent arteriole will further break off into capillaries, and this set of capillaries is known as glomerulus, which is present in this cup-shaped structure called Bowman's capsule, right? So set of capillaries, these are capillaries in the Bowman's capsule, a specific term given here. And when the capillaries will rejoin to come out, that is efferent arteriole. So do you see that afferent arteriole is broader while efferent arteriole is narrow? So this will lead to the pressure development in the glomerulus, because of which everything is going to be filtered out except for the blood cells and the proteins. Is that clear? So Bowman's capsule is this cup-shaped structure. Do you see this PCT? Do you see this U-shaped structure called loop of Henle? The part which is carrying the filtrate downwards is called the descending loop, and then the part of the filtrate carrying it upward is known as ascending limb. Is that clear? Then it gets changed into DCT again, PCT and DCT. PCT is a place where maximum reabsorption is going to occur. In DCT, it will be facultative reabsorption or conditional, only if required, otherwise not. While least reabsorption will take place in Henle's loop. Least reabsorption. Minimum. Are we clear? Right? And then so many nephrons will open into a single collecting duct. So collecting duct is not a part of a particular nephron, right? Then do you see then this efferent arteriole? See, look at the blood supply. First, renal artery breaking into efferent arteriole, forming glomerulus, then rejoining to form efferent arteriole, further breaking down around the PCT and DCT into capillaries, and then forming the vasa recta. These are peritubular capillaries. And again, breaking down around the Henle's loop to form vasa recta. So capillary network around loop of Henle is known as vasa recta. Are we clear? Right? So this is a Malpighian body. So Bowman's capsule, cup-shaped structure, and the glomerulus. This is the glomerulus, right? Together both of them are known as renal corpuscle or Malpighian body. Is that clear? So we see efferent arteriole broad, efferent arteriole narrow. Please let me know these points are clear. Moving on the next diagram. Very important diagram, and this diagram summarizes everything, that which part is showing reabsorption and tubular secretion of which component. Now see, so this is a PCT, this is a PCT. So first of all, see, cortex area carries what all parts of nephron are present in cortex. It contains Bowman's capsule, PCT and DCT. Medulla carries the loop of Henle. Is that clear? Now, in the PCT area, this is the PCT area, this is the DCT area. In PCT, the arrowhead moving away from the nephron shows reabsorption. This is reabsorption. And the arrow towards the nephron shows the tubular secretion, right? Which components are being reabsorbed and which are being secreted? You should take notice of this and remember it as such. From loop of Henle, we see that descending loop takes out water, right? So that is why descending limb was known as the diluting limb. It is permeable to water, impermeable to electrolytes, while the ascending limb is permeable to sodium and impermeable to water. And because sodium is coming out, hence it is concentrating. Sorry. So this one is, this one descending, because the water is coming out, that is why it is concentrating limb. While in in the ascending limb, sodium chloride is coming out, so it is permeable to electrolytes, hence it is the diluting limb. Is that clear? So it is permeable to, ascending limb is permeable to electrolytes. It is permeable to electrolyte. Very important line, already a PYQ. While the descending limb is permeable to water. Concentrating limb. Is that clear? Please let me know all these points are clear. Yes? And also, do you see which are the two main components in medulla which are resulting in development of osmolarity? What are the two chemicals which help in development of osmolarity as you go deeper into the medulla? One is sodium chloride and second is urea. These two help in the generation of higher concentration, right? All right, guys. So next, this is the counter current mechanism and something that I've already explained. See, as you go deeper into the medulla, the osmolarity goes on increasing from 300 till 1200. So this is the descending limb. The descending limb is descending loop of Henle. This is permeable to water. Water is coming out, so the osmolarity is increasing from 300 to 1200. So as the filtrate moves into the ascending limb, it becomes permeable to sodium chloride, right? And again, once again, it starts becoming dilute. When sodium chloride will come out, it will start becoming dilute again, right? So all this which is coming out from the walls of the nephron, walls of the this loop of Henle, it is going into this interstitial fluid, and through interstitial fluid it is moving into the vasa recta. This is all vasa recta, right? So see the pattern in which things are moving. This is vasa recta and nephron, and hence represents a counter current mechanism. In counter current, there is opposite flow of things, right? So in descending, see, there is opposite flow in descending descending loop of Henle and ascending loop of Henle. Filtrate going downwards, filtrate going upwards. There is counter current in the vasa recta, blood going downwards, blood going upwards, and between the adjacent limbs also. See here, in the descending loop of Henle and ascending, in the descending loop of Henle and ascending vasa recta, do you see there is opposite flow? This is ascending vasa recta. Is that clear? So counter current is applicable, or it is observed at 3 different levels. In the 2 limbs of loop of Henle, in the 2 limbs of vasa recta, and the adjacent limbs of vasa recta and loop of Henle. So that is counter current mechanism, and this helps in concentrating the filtrate. So this counter current mechanism is only present in mammals. Is that clear? This concentrating mechanism is only in mammals. All right, guys. So with this, we complete another chapter of human physiology, and let's move on to movement and locomotion and come on to the diagrams. First diagram is that of cross-sectional view of muscle. So outer structure. So this whole structure represents the muscle. In muscle, guys, we have muscle bundles. So these are the muscle bundles or the fascicles. These are the muscle bundles. Within the muscle bundles, 15 minutes. Yes, guys, let's continue. So this is a skeletal muscle, let's say. Muscle has muscle bundles. So inside muscle bundles, we see these muscle fibers. So inside the muscle bundles, so this is level 1, this is level 2. Inside the muscle bundles, we see muscle fibers. The muscle fiber is equivalent to the muscle cell, right? So all the vocabulary that you use for a cell, you are going to use the same terminology here also. So plasma membrane is known as plasma lemma, just that we are going to use the prefix sarco. So the plasma membrane of muscle fiber is known as sarcolemma, right? Sarcoplasm, sarcoplasmic reticulum, and so on. So this is muscle fiber, and they are all surrounded by these blood capillaries. Is that clear? So muscle bundle, also known as fascicle, and then these muscle fibers. Is that clear? And within the muscle fibers, we'll have myofibrils, then myofilaments, right, which will actually help in contraction. Are we clear? Now this structure represents the unit of contraction, which is sarcomere. This is a sarcomere. See, between two Z lines, this is the EM, structure, and this is the diagrammatic way of representing the same. So this is Z line. These are the Z lines. These are the Z lines, right? Between two Z lines is present a sarcomere. Now sarcomere contains one, so light band as well as dark band. Is that clear? What is basically light band? Light band, also known as I band. So see, this is the light band. Why do we call it light band? Because light can penetrate through it. Light can penetrate through it. Is that clear? Now, just a second. Are we clear here? Yes, guys. So this is the light band. Light can easily penetrate through this. So Z line is actually dissecting the light band or the I band. Yes, can we say that? Z line bisects the I band. Right? So when we say sarcomere is between the two Z lines, so that means there is half I band on this side and half I band on the other side, and in between we are going to have the A band, right? Half I band on this side, half on this side, right? And in between an A band. So A band depends upon the length of the thick filament. So as far as the thick filament extends, it is a A band, also known as dark band. So this is the dark band here, and this is the dark band here. Clear? So light band, guys, is made up of, light band consists of only thin filament. Very important. What does light band consist of? Only thin filament. While the dark band contains thin filament as well as thick filament. It contains both thin plus thick filament, right? Now, the thin filaments are attached to the Z line. What about these thick filaments? These thick filaments, they are not hanging in air. They are also attached to this line present in the center. So this line present in the center, is that clear? Present in the, all right guys, just a second. Are these points clear till here, everyone? Please let me know, are these points clear about the Z line and all this? Are we clear till here? All right, let's proceed then. All right, so in the center of A band lies an M line. So thick filaments are attached to this M line. Clear? So in the center of the A band, we have this M line. Now what is this H zone? Do you see that as far as this A band is extended, there is some overlapping of thin filament from the outer side on the peripheral area? Yes. So in A band, there is thin filament on the peripheral area and thick band extends throughout. Now, while there is a small area in the center where the thin filaments are not present. So this area in the center, which is not overlapped by the thin filament, is called the H zone. So this was a PYQ. What is H zone? It is a centrally placed area, central area where only thick filament present. Central area of A band where only thick filament is present. Please tell me these points are clear. Right? So you should know the definition of all these points. And now let's look at the structure of thin and thick filament. Yes. So see, the thick filament, guys, thin filament is made up of three proteins. Which are the three different proteins? Actin, tropomyosin, and troponin. How many components of each are present? So first we have F-actin. F-actin means filamentous actin. There are two filaments of F-actin in the helical manner. Then two filaments of tropomyosin, again helically bound. And then there are three subunits of troponin. So three subunits they collab together to form one troponin, and these troponin are dispersed at regular intervals. Do you see? So three different proteins constitute your thin filament. Is that clear? All right. Now basically on the F-actin, guys, we have myosin binding site. We'll have myosin binding site here. Right? We have myosin binding site here. Now this is your myosin monomer or meromyosin. This is a monomer of the thick filament. So such 300 to 400 units will come together to form a thick filament. So see, each meromyosin consists of this tail, then arm, this is the arm, and it has a head. Head has two sides. One is actin binding site and then second is ATP binding site. ATP binding site is the place where ATP breakdown will take place to release energy because muscle contraction demands energy. So the myosin head has actin binding site while F-actin has myosin binding site. Is that clear? Now see here. Let's look at the process of muscle contraction with the help of this diagram. See, so first of all, when ATP breakdown occurs, ATP breakdown or ATP is hydrolyzed and it releases ADP and phosphate, then this myosin head will bind to actin, right? Once the myosin head will bind to the actin, we say cross bridge formation has taken place. Is that clear? Cross bridge formation represents the beginning of muscle contraction, right? So basically ATP breakdown, release of ADP, and then ADP and phosphate is produced, then cross bridge is formed, and finally what will happen when ADP will come out, this myosin head which has bound to the thin filament, it will move backwards. So there is backstroke actually. It will move on the backside, and this leads to sliding. So what will happen? The head which is bound to the thin filament, it's like this. When the head will move this way, this is going to lead to sliding or rotation of the thin filament. So thin filament will slide. The thick filament will never slide. Thick filament will remain the same. Is that clear? And after that, if we want the muscle to relax, then ATP regeneration should take place. So when ATP comes back, there is breaking of the cross bridge and the muscle relaxes. This leads to muscle relaxation. This is the beginning of muscle contraction. Is that clear to everyone? All right, guys. Now moving on to, so in this diagram we see that how sliding occurs, and during the sliding, guys, see sliding occurs in sarcomere, right? During the sliding we see Z lines come closer. They come closer. Yes, from here they have moved here. So they come closer. I band will also reduce. H zone will also become smaller, or it also reduces. One thing that will remain the same, that is the A band. A band is going to remain the same. Is that clear to everyone? All right. Next, moving on to the skeletal structure. So this is the human skull. Look at this location of each bone. One forming forehead is frontal bone, then parietal bone, temporal bone. How many of them are present in our skeleton? And occipital bone. Right? And we have two occipital condyles, hence our skull is called dicondylic skull. Clear? Look at the location of the condyle here. Then this is sphenoid bone, again a part of the cranial bones. All these blue highlighted, these form the cranial bones. How many cranial bones are present? 8. Right? Sphenoid and ethmoid. Then rest of the bones not highlighted here, that is lacrimal bone, nasal bone, zygomatic, the cheekbones, maxilla, upper jaw, mandible, lower jaw. These constitute your facial bones. They are not all here, not shown, not all of them are shown here. These are 14 in number, 14 facial bones. So 14 facial and 8 cranium total constitute the skull bones, while here only few of them have been showcased. Hyoid is not a part of skull. Remember that. Hyoid is not a part of skull. Not a part of skull. Is that clear? This is an independent bone present beneath the this buccal cavity, and it is horseshoe shape, only one in number. Clear? Then this is your vertebral column, S-shaped structure divided into 5 regions, guys. We have cervical, thoracic, lumbar, sacral and coccygeal. How many? 7 vertebra in the cervical area, that is 7 cervical, 12 thoracic, 5 lumbar, 5 fuse to form 1 in the sacral area, and 4 fuse to form 1 in the tail area, that is coccygeal. Is that clear? Now these are your rib and the rib cage. So let's talk about the ribs. There are 12 pairs of ribs, that is 24, out of which 7 pairs are true ribs, 3 pairs are false ribs, and 2 pairs are the floating ribs, right? True ribs, true ribs are going to attach, they will be, these first 7 ribs, they will be attached directly to the sternum through the haline cartilage, through this haline cartilage. Then 8th, 9th and 10th will not be directly attached to the sternum, but will attached via 7th vertebra, again through the haline cartilage. And then we have 11th and 12th. What happened? Okay, 11th and 12th pair of ribs, these are floating. Why do we call them floating? Because they are not attached to anything on the ventral side. Is that clear? These are floating ribs. Are we clear? Yes, guys. So overall, our ribs are bicephalic. Clear? Now, then we have our rib cage. So rib cage overall is formed by 3 things. Sternum on the ventral side. This is the sternum. This is on the ventral side. Then vertebral column on the dorsal side. And our ribs. These 3 together form the rib cage. Are we clear till here? All right. Now let's talk about the pectoral girdle and the upper limb. So see, our pectoral girdle is formed of scapula and clavicle. So one on each side. So in total we have 2 scapula and 2 clavicle. So scapula has this cavity called glenoid cavity. This cavity is present in the scapula, and to this cavity the head of the humerus is going to fit. This is the head of humerus. So head of the humerus fits into the glenoid cavity to form ball and socket joint, right? Ball and socket joint. Is that clear? So this is humerus, then 1 radius, 1 ulna. How many carpals? We have 8 carpals, we have 5 metacarpals and 14 phalanges. So this is the number and the order of the bones present in our forelimbs. So in total 30 bones are present in your forelimbs, each forelimb. So in total 60. Let's see the hind limb also. So hind limb has again 30 bones. Now hind limb, that means your leg overall, is attached to the hip girdle. Hip girdle is made up of 2 coxal bones, 1 on each side. So this is 1 coxal bone, right? Coxal bone is further formed by the fusion of ilium, ischium and pubis. So where ilium, ischium and pubis fuse, they form this structure called acetabulum. They form the structure called acetabulum. So they will fuse to form acetabulum, and it is in the acetabulum that head of humerus will fit into. So acetabulum plus head of femur, again they will form the ball and socket joint. This is patella, that is the kneecap, and again 1 tibia, 1 fibula, 7 tarsals, that means your ankle, then 5 metatarsals and 14 phalanges, again 30 bones. Is that clear here? Now, very important part, that is your joints, but there is no diagram related to this. Please make sure that you learn these examples. Last second chapter, guys, let's quickly go through the diagrams of this chapter in neural control and coordination. See, this is the structure of a multipolar neuron. So these are dendrites. There are Nissel's granules present in dendrites as well as in cell body, right? This is the nucleus, and from the part where which is surrounded by Schwann cell, this is from where axon begins. Now Schwann cell will give rise to the myelin sheath, which is a fat-rich layer, and therefore this neuron will be known as myelinated neuron. So what is node of Ranvier? It is that place where Schwann cell is present but myelin sheath is absent. Now this is important for quick conduction of the impulse. The last part is called axon terminal, and we have these circular structures called synaptic knobs which contain synaptic vesicles containing the neurotransmitters. Is that clear? Now let's move on to the structure of synapse. See, this represents the structure of synapse. So synapse is formed of three components. One is the presynaptic membrane, second is the synaptic cleft, that is the space, fluid filled space, and the postsynaptic membrane. Now in case the synapse involves a neurotransmitter, it is called a chemical synapse. So this figure represents a chemical synapse. So these neurotransmitters are released by synaptic vesicles. Neurotransmitters are present in these synaptic vesicles. Synaptic vesicles will fuse with the presynaptic membrane, release them in the synaptic cleft, and then the receptors for these neurotransmitters are present on postsynaptic cleft. So these are the questions, where are receptors present? Is that clear? So receptors for neurotransmitters are present on the postsynaptic cleft. Is that clear? Let's quickly go through the structure of human brain, guys. This is sagittal view. So there are three parts: forebrain, midbrain, and hindbrain. Forebrain further consists of cerebrum, the largest part, highly folded, consists of two cerebral hemispheres, and on the inferior side it is joined together by the corpus callosum. Is that clear? So this is the corpus callosum here. This is the corpus callosum. Now beneath that we have thalamus and hypothalamus, which is a part of diencephalon, right? So, and this is hypothalamus, and this represents a pituitary gland, which we are going to discuss in the next chapter. Clear? Then below the diencephalon, or basically surrounded by the forebrain, we have this part called midbrain, right? Do we see different regions of hindbrain here? This is the pons veruli, this is the cerebellum, and this represents the medulla oblongata. And from medulla oblongata starts the spinal cord. Now there is a special duct which runs through the midbrain, which is called the cerebral aqueduct. Is that clear? All right, guys. So with this we complete this chapter, and last not the least, let's look at the diagrams of human endocrine system. These are the different glands. So on the ventral side, guys, we have this pituitary gland, and on the dorsal side we have the pineal gland. I've just shown you the structure and the placement of pituitary gland, right? Then this gland is thyroid gland, and on the dorsal side of thyroid gland itself we have four parathyroid glands. This is bilobed thymus, this represents leaf-shaped pancreas, and above the kidneys are present adrenal, and these are the gonads, ovaries in females and testes in case of males. So look at the diagrammatic representation showing the relationship of pituitary and hypothalamus. This is hypothalamus. These are hypothalamic neurons, right? Hypothalamic neurons are releasing the hormones. So these hormones are released in the portal circulation, will be carried into the anterior pituitary, right? So what kind of hormones are coming along the portal circulation? Releasing hormones, inhibitory hormones, while the other neurons which have their axonal endings opening in the posterior pituitary, they are going to release oxytocin and ADH. So oxytocin and ADH are basically produced by hypothalamus but released by posterior pituitary. Posterior pituitary doesn't produce its own hormone. Are we clear? So see the structure of thyroid gland. This is the thyroid gland present around our vocal sac. right? This is the trachea, and hence this is a bilobed gland. So this is H-shaped or bilobed butterfly-shaped gland, follicular in nature and connected by a small isthmus, right? And on the dorsal side, do we see these 4 parathyroid glands? So on the dorsal side, we see this is on the dorsal side of thyroid. So a gland within the gland. Clear to all? So this is kidney upholding adrenal gland. So that is why we call it suprarenal gland also. Outer region cortex secreting corticoids of 3 different types, and medulla secreting the emergency hormones or the catecholamines. Clear? And here we have this very important diagram showing the functioning or the mechanism of hormone. So see, this hormone is binding to the receptor present on the membrane, right? So this hormone has a extracellular receptor. So which all hormones will have extracellular receptors? All the protein hormones, proteinaceous hormones, amino acid derivatives, all these hormones will have extracellular receptors. Is that clear? Okay. Now, if the hormone is binding to the receptor on the membrane, so in order to carry the message within the cell, we need some second messengers here, like cyclic AMP, calcium, IP3, inositol phosphate, which will lead to biochemical responses and eventually the physiological responses. And then, guys, we have iodinated hormones, iodine carrying hormones, that is T3, T4, thyroxine, tetraiodothyronine and triiodothyronine, all the iodinated hormones and steroid hormones. These 2 type of hormones will have their receptor within the nucleus. Their receptor is present, receptor within the nucleus. So because the hormone is going to cross the plasma membrane itself, intracellular receptor or intranuclear receptor, because the hormone is itself going to cross the plasma membrane and go into the cell, carry the message within the cell itself, so we don't need any secondary messenger. No secondary messengers are needed here. So see, the hormone and receptor complex is formed within the nucleus. That's where genetic changes will occur and physiological responses will take place. Are we clear, guys? So with this, we complete all the diagrams of class 11. Is that clear? All right, guys. Now, tomorrow I'm going to take a very important session, guys. Very important session at the same time, in which we are going to revise some last minute topics. I know all the things are already in your head, but just there are certain things which need repeated revision, right? The fact-based or some difficult things. We are going to revise those things tomorrow. So make sure that you join me tomorrow 100% at the same time, 12 p.m. We are going to do that final revision tomorrow. Is that clear? Don't forget to join me tomorrow. And once again, wait a second. Yes. So once again, guys, please make sure that now at least you are definitely taking this Unacademy All India Test Series and attempting these tests every day. Not even a single day should be left where you are not practicing these questions. Is that clear? Not even a single day should be left when you're not practicing these tests. Time bound, complete syllabus coverage. After these tests, now analyze which topic is left and revise only that. This is a very crucial time. Last few days, we are only going to hit on the target, the difficult topics, and how will you identify your own weak point? Through UAITS. So make sure that you take this Unacademy All India Test Series for complete syllabus coverage and revise the whole syllabus. Test yourself at your convenience. Alright guys, thank you so much. I'll see you tomorrow once again at 12 p.m. with a very important session. Don't forget to join me. And we are practicing questions in special classes. So please make sure that you you attend all the special classes and don't miss any such question. We have revised the exemplar. We are doing other important questions from all the units. Thank you so much. I'll see you tomorrow. Ta-da!